before?
No, it's the first time, actually. Yeah, it's very nice, actually. I really enjoy it.
It's grown enormously.
Yeah, enormously, yeah. So how many participants?
1,000 plus this year, and of that, 450, I think, gave talks or
yeah, the
posters
workshop
so
so it's
it's a big participation.
yeah. Yeah, and people discuss about that all the worldwide. I mean, in many different fields that are not just only computational and system, right? It's just spreading more and more.
Yeah. Hopefully people find an opportunity for collaboration.
Yeah, exactly. Jeez, this light, I would become blind.
No, that. It's funny, I've only been sitting there.
Yeah,
so
exactly. I hope so. Well, I will be just totally blind afterward.
Yeah. Oh, actually, we're supposed to start. It's 2, I guess.
Yeah,
okay, good. All right, I know some people are still coming in, but it's a couple minutes after 2, so we should go ahead and get started. We have another wonderful session coming up, and leading us off is Camilla Bellone from the University of Geneva. So welcome.
So thank you to the organizer for inviting me. Thanks to you to be here after the lunch. So today I will present some unpublished data trying to solve a very fundamental question. So how we decide whether to interact or to avoid another individual. So we all know about the importance of social behavior in our life, and actually whether we found ourselves in the office with colleagues, in a bar with friends, or even if you have illusion to walking alone in the street, we are constantly surrounded by social cues, and we need to integrate these social cues with a context in order to adapt our behavior. If this does not happen correctly, we may end up to develop maladaptive social behavior, social dysfunction, as happen in psychiatric disorder or in some situation of daily life in our society. As I say, social interaction are very important, and we all consider social interaction more or less rewarding. My lab and other laboratory in the world has contributed in the last 5 to 10 years to really dissect the neurobiological mechanism underlying social reward processing. But we also know, unfortunately, that social interaction are not only rewarding, but that in our life we may experience social interaction that are indeed aversive. For example, we may be aggressed, children can underline bullying. So we have this dichotomy of social interaction that can be highly rewarding experience, but as well aversive. So what happened when we meet for the first time an individual in the street? So we first have to understand whether the individual that is in front of us could elicit with us an experience that is rewarding or aversive. And then, depending on this first experience, we need to update the value representation of this other individual and fast enough decide whether to approach or to avoid. So for the talk of today, I would like to just pose one definition, that is how we define social valence. And for us, social valence is really the degree to which the experience with a conspecific could be pleasurable, so rewarding, or aversive. And the question we would like to answer is that how does an individual process social valence and learn to adapt the behavior depending on the past experience? So we use mice as animal models, and we observe animals most of the time in freely moving interaction. And now I will present you one of the videos where you could appreciate 2 mice. So these are 2 male mice that are unfamiliar one to each other. And you can observe in these slow motion videos the quality of interaction that you probably would define as rewarding. So mice explore each other, they have a sniffing behavior, they have some active and some passive interaction. This interaction is qualitatively very different from these other videos, where you have this mouse that is taking care of himself and has been attacked by the other mouse. And you can immediately see that qualitatively, even if maybe the time of interaction is exactly the same, the quality of his interaction is dramatically different. So how can we quantify this different aspect? So we had to set up a very simple experiment where we observe for 15 minutes the behavior of two unknown mice, and we use male-to-male interaction as a first attempt. So in this free social interaction paradigm, we use always object as a neutral control, and then we use specific sex-matched juvenile that elicit more approachable and rewarding interaction. And we use aggressive CD1, so very aggressive strain of mice, as the negative and aversive experience. And I want to specify that the CD1 is not just the strain, it's because these mice are really trained to be aggressive. So of course, the first thing that we can have a look at is the proximity to the stimulus and the time spent in this proximity. So how much time they spend interacting with each other. And you can immediately see that the mice generally spend more time interacting with the juvenile mouse than with the CD1. But this aspect did not catch the qualitative aspect. So we decided in this first attempt to really look at 4 different types of reciprocated behavior. So first one is the reciprocal interaction, where there is a nose-to-nose interaction between the two mice. The approach, so from the perspective of experimental mouse when he's approaching the stimuli. And you can see here that in the juvenile experience, you have high percentage, long time spent in these 2 types of interaction, while with the CD1, you have a lot of bout of aggressive behavior and avoidance behavior. So based on this very, very simple task, what we can really think that what we are looking during the juvenile experience is a rewarding interaction characterized by affiliative bout. And the experience with a CD1 is an aversive experience characterized by antagonistic interaction. Now, the question is that where in the brain is computed the social valence? So there are many different regions that in the brain has been associated to social behavior, and actually would be interesting to find a region that has not been associated to social behavior. You have a region has the cortical cortex, so anterior cingulate cortex, prefrontal cortex, that are really important for the reward value assignment during social interaction. You have region like the amygdala, you have a region of hypothalamus. And one circuit that we have focused attention on in the last years is the reward system. I told you the social interaction are rewarding, so we focus a lot of attention on reward system. So this system originates in the ventral tegmental area, where there are these dopaminergic neurons that project to the ventral part of the striatum, the nucleus accumbens, or to the prefrontal cortex. And actually, back almost 10 years ago, in the lab of Carl Dijsselhoff, Lisa Gunedin showed that the activation or inhibition of dopamine neuron were able to bidirectionally regulate social interaction. So activation of dopamine neurons increase social interaction, and inhibition decrease social interaction. Then recently, we have used tetrode recording in vivo in freely moving mice. And what you could see here is on the left, the position probability of our experimental mouse in function of the other mouse, and plot the normalized frequency of the dopaminergic activity. And what we could really observe was that there was an increase in activity of dopamine neurons when the stimulus mice was in the frontal field compared to our experimental. Moreover, what we have done also recently is set up a behavioral paradigm that was really able to show the rewarding aspect of social interaction, and in particular the motivational aspect. So in this task, also in this case, you have, okay, the videos here doesn't work, doesn't matter. So here you have a cage where this is our experimental mouse, and he has to press a lever in order to activate the guillotine door that open, and the door stay open for 7 seconds. And then the mouse, if he wants to interact again with the other with stimulus, he has to press again and again. And so what we have shown is that mice are able to learn this task. They show operant behavior. There is an increase in number of lever press. And moreover, what we have observed when recording the activity of dopamine neurons during this task is that dopamine neurons were encoding prediction error signal. Indeed, before the learning, there was an activation of dopamine neurons during the interaction window, so when the social stimuli was present, while after the learning, the activity of dopamine neurons shifted at the lever press. So there was a prediction of a reward coming, and actually, if we were omitting the social stimuli on the other side, there was a dip in activity, really indicating there was a social reward prediction error. And in this paper recently, what we have shown is that this prediction error signal is really instrumental for social learning process. Now, the activity of dopamine neuron is important, but also what is even more important is that dopamine is a neuromodulator that acts onto the nucleus accumbens. And actually is a nucleus accumbens that is a part of the ventral striatum that has been shown being really fundamental for social reward processing. So in this region, we have the majority of neurons that are medium spiny neurons that are inhibitory neurons containing D1 or D2 receptors, so receptor for the dopamine. They receive a lot of excitatory input from different brain regions, and the dopamine acts as a modulator. So one important aspect is that people have shown that the nucleus accumbens and the reward system are activated by rewarding experience, but also by aversive experience. So our initial idea was to really understand whether, in the context of social valence processing, the nucleus accumbens, and in particular the D1 neurons of this area, were responsible for compute the social valence, positive and negative. So the role of nucleus accumbens social interaction, as I said, has been shown by different laboratories, and mine was also one of them. What we've shown in this experience is that when we are recording the activity of medium spiny neurons in the nucleus accumbens using fiber photometry, we were observing that when the mouse was in close proximity of another stimuli, there was an increased activity of these neurons compared when the mouse was in close proximity of an object. Moreover, in the same paper I showed you before by Dijkstra's lab, what they have shown is that the activity of D1 containing medium spiny neuron that was important for processing social reward, because when D1 were activated, there was an increase in social interaction. So the nucleus accumbens is a complex structure per se, and there are different subdivisions. And one subdivision that immediately attracted our attention was the mesolimbic lateral part of the shell, because this part of the nucleus accumbens were really involved both in rewarding and aversive experience. So we decided to focus the attention on this lateral part of the nucleus accumbens and see how D1 medium spiny neuron were computing rewarding and aversive social interaction. So how we did that? So first of all, we used D1 Cre mice, so the Cre under the promoter of D1, and we injected the GCAM, and we implanted the miniscope. So this allowed us to record from the neuronal population, and we recorded the activity of these neurons during the neutral experience, during rewarding experience, and during aversive experience. It is important to say that each mouse was underlying the 3 different experiences one day after the other. So overall, at the population level, you can see that there were no increased activity during neutral experience, but there was an increase in activity during the rewarding experience and during also the aversive experience. We looked then at each neuron independently, and what we were able to calculate is the percentage of neurons that were both activated during positive and negative experience. So around the 70% of neurons are both activated by the juvenile or by the CD1 experience. Then we look at the activity of each neuron in function of a relative position of a stimulus and experimental mouse. Here we put at the center our experimental mouse and look at the activity of a medium spiny neuron in function of a position of a stimuli. Here you can see that there is an increase in activity of D1 medium spiny neurons during both rewarding and aversive experience, and that this activity was really when the mice were in close proximity. Here is the quantification of the data. You can see that both for positive and negative experience, rewarding aversive experience, you have an increase of activity of D1 neurons during the closer they were. So these data were really suggesting that the D1 neurons were computing the proximity of a stimuli independently of the social balance. Now, the question we ask is that how these neurons are activated. So which are the excitatory input that contribute during this rewarding or aversive experience to the activation of this neuron? So in order to answer this question, we use the FOSCRI-ERT2 mice, we inject the flex retroviruses, and then what we were able to expose the mice either to a CD1 or a juvenile, and then we were able to see which were the neurons that were projecting onto the medium spiny neurons of the nucleus accumbens, and that were active during both rewarding and aversive experience. Since the NAC were also activated by both positive and negative, we really want to see which were the input onto this region that were activated also independently of the valence. So among the different regions that we have looked at, one that catch our attention is the anterior insular cortex, where we found that there was neurons that were activated by the juvenile or the CD1 experience. Interesting enough, and I will make a long story short, we characterize the nature of these neurons of the anterior insular cortex that were projecting onto the nucleus accumbens. And by doing RNA scope, we found that these neurons were also containing D1 receptor. So this was the first surprise that there was a specific connection between D1 containing neurons in the anterior insular cortex that were projecting down to D1 neurons in the medium spiny neuron of the ventral striatum. Now, the question was, okay, so if we do the same experiment as before, we're now recording from anterior insular cortex projecting down to the nucleus accumbens, we're using miniscope, what we see, what is the anterior insular neurons encoding during this experience. So, in this case, we injected G-camp retrogradely in the nucleus accumbens, and we implant a miniscope with a grid lens into the anterior insular cortex in D1 Cre mice, so specific D1 neurons projecting down to the ventral striatum. And we did exactly the same experiment as before, so we exposed the mice to a neutral stimuli, to a CD1, to a juvenile mouse, or to a CD1. You can see here at the population level, there is an increased activity of these neurons. But already when we look into the population of neurons, single neuron level, and see which were the percentage of neurons that were really activated by both experience, we saw that we were down to 50%. Anyway, there was a 50% of neurons that were also activated by positive and negative experience. We then again plotted the same type of analysis, the activity in the relative position, but here the situation was much more complicated because neither for the rewarding experience or aversive experience, there was a real precise time lock event that we could reveal. So you can see there is somehow higher activity in the close proximity for rewarding experience, but was much more scattered for aversive experience. And in any case, when we were quantifying this data, we could not see any significant difference and correlation between the activity and the proximity. So this data was really suggesting that the activity of anterior insular cortex were not encoding the proximity of a stimuli. So why and when were these neurons activated? In order to answer this question, what we did is that we trained a variational autoencoder using the interaction observed during the juvenile experience. And then we built the contact classifier using a support vector machine, and we were able to create a behavioral classifier that was somehow predicting the social valence. So in this case, our classifier, what was able to predict was at 90% almost to have affiliative type of interaction during the juvenile experience, of having antagonistic interaction during the CD1 experience. So then we took the data from the calcium recording, we took the raw signal, we extract some feature, and we made the classifier out of a calcium imaging predicting the behavioral valence. So what these data were telling us that based on the calcium activity, where we're predicting to have affiliative contact during the juvenile experience, and we were predicting antagonistic contact during the CD1 aversive interaction. So that was meaning that anterior insular cortex were not really predicting and encoding the proximity of the stimulus, but was somehow encoding the relief valence. So how this is possible, how this may occur? So what we did is that we used the Fourier analysis on the calcium data, and what we're able to reveal is that during interaction with the juvenile, so during the rewarding interaction, there was an activity of medium spiny neurons that was on the low frequency band, while during the aversive CD1, the same neuron were shifting and were be activated by a higher frequencies with higher frequency. So that was very intriguing for us. So what we were able so far to show is that activity within the anterior insular cortex neuron projecting to the ventral striatum were predicting the valence of a social experience. And moreover, that the neuron within the anterior insular cortex were firing at low frequency during the rewarding experience and firing a higher frequency during the aversive interaction. So now, what happened? So if you think when an individual meets another individual, it means that there are probably different frequency stimulation of neurons in the anterior insular cortex, depending on the valence, whether the experience is rewarding or aversive. But our question was, is this activity sufficient to leave a trace within the nucleus accumbens such that we change the value representation and we are able, thanks to these traces, this memory trace, to approach and avoid the individual the second time we meet them. So our question is that is able this activity to generate a memory trace in order to instruct the approach or avoidance behavior. So in order to test this hypothesis, we needed first to verify whether even in mice, the first experience with a positive or negative mouse was sufficient to instruct approach and avoid behaviors on the second day. So do the mice remember and recognize the conspecific with whom they interact the day before? So on the first day, they have a neutral experience, they have a juvenile or the CD1. And on day 2, basically we design an approach avoidance test on a corridor where first the mouse is free to explore. Here there is an encounter. And then in the post-test, for 10 minutes, we place in the encounter the same exact stimuli that were exposed the day before. So we look at the interaction time and the proximity, and actually it's interesting to see that for the object, there is no difference between the pre-test and the post-test. As expected, the objects are not very interesting, but the mice were approaching and spending more time with the juvenile mouse, the one with whom they had a nice and positive rewarding experience, but they were really avoiding the mice with whom they had this aggressive and aversive experience. So behaviorally, we were fine. We had a behavior that could reproduce and show approach and avoidance behavior and sort of learning and memory. Now, here's a social valence store at synaptic level, and can we observe a valence-dependent form of social plasticity that may instruct this approach and avoidance behavior? We know memory traces mean synaptic plasticity, so we really hypothesized that maybe at the interior insular cortex to nucleus accumbens, the social experience was generating a form of plasticity that were depending on the valence, and was this form of plasticity that was the trace, the memory trace that is needed in order to approach and avoid. So to test that, we moved to slice physiology. So first we had to prove that this connection between anterior insular cortex and nucleus accumbens was functional. So what we did is that we injected the chanorodopsin in the anterior insular cortex in D1 Cree mice, and we inject a flex stem cherry in the nucleus accumbens. So we were able really to patch clamp from D1 positive neurons, and look at the synaptic connection between these two regions. So we performed pair recording, and we first could observe that indeed there is a preferential projection of D1 neuron in the insula to the D1 neuron in the lateral nucleus accumbens. And then we say, okay, there is a form of plasticity that is generated depending on the experience. So on the same setting, we expose first the mice to an object, to a juvenile or to a CD1. We place back the mice in their own cage, and 24 hours later, to leave the synaptic plasticity be induced and expressed, we cut the slice and look at excitatory synaptic plasticity. So the first proxy that we look at in change in the ratio between the two glutamatergic receptor, AMPA and MDA. So the molecular mechanism is not needed for this audience, but what is important to show is that this is really proxy of a strengthening of the synapses. And actually what you could see is that after the juvenile interaction, so 24 hours the interaction, you have an increase in AMPA and MDA ratio at this anterior insular cortex to nucleus accumbens synapses. But this was only specific for juvenile, but not for the CD1, neither for the neutral experience. Can the CD1 experience generate a different type of plasticity? We know that the plasticity can be expressed as a change in ratio between a glutamatergic receptor, but we know that also the quality of a transmission can change, and the type of receptor may change as indication of a form of plasticity occurring at that synapses. So we look for the presence of GLUA2 lacking AMPA receptor that are typical AMPA receptor that are unconventional because they flux the calcium and not only the sodium. And actually what we were surprising to see is that the CD1 experience, so 24 hours after the CD1 experience, this type of non-canonical AMPA receptor were expressed at that synapses, and only after CD1 interaction and not after the juvenile. While this is peculiar is also because this type of receptor, this non-canonical AMPA receptor, are often associated with aversive experience. So now, basically we have that social experience, depending on the valence, whether it's positive or negative, induce at the same synapses two different forms of plasticity, with an increase in AMPA-NMDA ratio that is expressed after the juvenile experience and an increase on non-canonical AMPA receptor after the CD1. The question was, is this induced because of different frequency of activation of anterior insular cortex? And remember, I showed you before that from our calcium analysis, we were really showing that during a juvenile experience, the neuron in the anterior insular cortex were firing a low frequency, while the neurons were firing a higher frequency during aversive experience. So our hypothesis is that maybe these two forms of synaptic plasticity are generated and induced by different frequency during the experience. So how to prove the causality between activity and the plasticity? So in order to do that, we use in vivo optogenetic stimulation. So we injected the optogenetic tool, so the channel rhodopsin, in the anterior insular cortex, and then place the stimulating electrode in the nucleus accumbens. In this way, we were able to stimulate only this pathway. And what we were doing is that in the control side, we use the 1 Hz stimulation, while, 1 Hz stimulation, 10 Hz stimulation, or 0.1 Hz stimulation, always in the contralateral side as the control. So 1 Hz, we were thinking would have emulate juvenile interaction, and the 10 Hz would have emulate the CD1 interaction. And really, the mice were placed in a new environment with no other stimuli than just the optogenetic stimulation. Then the mice were back to their home cage, and 24 hours later, we cut slices and we checked for synaptic plasticity. So here what we were able to observe is that, as I predicted, the 1 Hz stimulation was sufficient to increase the proportion of AMPA-NMDA receptors, so was mimicking what was happening with the juvenile interaction, while the 10 Hz stimulation for 10 minutes was able to emulate what was happening during the CD1 experience. So really indicating that there was a causal link between the activity and the plasticity. Now, if you follow me until now, what I show you that there is a specific pathway between D1 neuron in anterior insular cortex that project to the D1 neuron in the nucleus accumbens, and then at the same the same neuron of anterior insular cortex can fire a different firing frequency dependently on the experience. 1 Hz low frequency stimulation during the juvenile experience and higher frequency stimulation during the CD1 experience. And this activity was sufficient to induce a form of synaptic plasticity. In one case, an increase in AMPA-NMDA ratio, in the other case, an increase in non-canonical AMPA receptor. Now, can we really prove the last causality experiment that the activity was really linked to the behavior? So it was really this plasticity causally linked to the approach and avoidance. So in order to prove that, this is the last experiment we have performed, what we have done is that we have injected channel rhodopsin again in the anterior insular cortex of D1 CRE-positive mice. We have placed the optic fiber in the nucleus accumbens and also injected them cherry because we were on the same animal that we performed the behavior. We then also perform a patch clamp experiment. So on day 1, we exposed mice to a juvenile conspecific, the rewarding experience. And what we did is that we imposed a high frequency stimulation of 10 Hz each time the mice were in close proximity. So each time the mice were in close proximity, we were ourselves imposing a 10 Hz stimulation that normally were not occurring. And on the second day, we first look at the behavior, and secondly, we slice the mice and look at the plasticity. So behavior. So this is a control group. So as we saw before in the other cohort of mice, we have an increase between the pre and the post-test if the mouse is a juvenile mouse. But when the juvenile mouse was coupled with a 10 Hz stimulation, so we were imposing a frequency that was not the natural one, we totally blocked this approach behavior. So we did not induce any avoidance, but we were able and sufficiently able to block the approach. Then we patch the cell, and so we look at the synaptic plasticity at the anterior insular cortex to nucleus accumbens, and what you could see is that the 10 Hz juvenile experience was not able to increase the AMPA-NMDA ratio as for the juvenile alone. And moreover, when we look at the non-canonical AMPA receptor, what you could see is that on average, there is no differences, but we had a group of mice, so one cell for each mouse, so a group of mice that basically has an increase in this percentage of AMPA receptor that was non-canonical. And when we made a correlation between the avoidance index, so how much the animal were avoiding the animal on the second day, and retification index, we saw a positive correlation, indicating that avoidance were strongly correlated with the presence of this non-canonical AMPA receptor. So now I hope I convince you today that there is this selective pathway where we have during positive rewarding interaction and low frequency activation of the pathway that impose an increase in AMPA-NMDA ratio that is able to instruct the animal on the second exposure to approach the other individual. While during the exposure with a CD1, a very aggressive mouse, there is an activation of this same pathway at 10 Hz, and this is sufficient to regulate the expression of this non-canonical AMPA receptor that instruct the avoidance. So we conclude saying that for us, the social valence is encoded within this anterior insular cortex nucleus accumbens and is depending really on a fine-tune of activity depending on the experience. So with that, I would like to thank the people that have contributed to this work. So in particular, Pedro Espinosa, that is a PhD student that conducted almost all the experiment, and Benoit Girard, that conducted all the experiment of the analysis on classifier and with the calcium data, and the funding, and you for your attention.
All right, we certainly have time for a few questions. Maybe as people come up, I have one myself. So do you have a sense of whether or not the activity in the accumbens is encoding sort of that valence, or actually the motor behavior involved in approach or active avoidance? If you did these in head-fixed animals, would they still show the same changes?
Yeah, I mean, in net-fixed animal, we don't know. So the one in the nucleus accumbens, definitely they code at the beginning, the day one, the proximity, and is independent on the valence. For the anterior insular cortex, to be honest, we have tried to time lock the activity of these neurons with many different types of the behavior, and we were not able to find any single specific behavior that was responsible for this activity. This is why we built the classifier, to try to look at it in a more unsupervised way.
Great. All right, we'll start over here.
That was really pretty. I'm wondering if you can sort of speculate more about the dynamics of the mechanism sort of in a real world environment. So if an individual is going to go out and he's going to hang out with his friends for a while, and then he's going to go hang out with some foes and defend his territory and then go back to his friends, how do you imagine these molecular results playing out? Are they in different boutons, or like, can you imagine?
It's hard to imagine and to speculate too much. I mean, I think that, of course, I think that an important role is played by the novelty of experience. So we move the mice from their own cage, we place in a different cage, so the context for sure play a role. But what I could imagine is that To be honest, I think that our baseline or the baseline of our mice in our cage is the juvenile. So the juvenile is the baseline. So there are a little bit of distortion of the baseline, and this increased AMPA-NMD ratio, these other things is probably the novelty of experience. Well, I think that what is really different from their baseline is the aggression of a CD1. This is something that they have never experimented in our experimental conditions. So I think that is why we really can change the positive experience with a negative one, but we were not able to do the other way around.
Yeah, thanks so much. I'm a little bit confused about the difference between an increase in AMPA receptors when you have the increase in the AMPA and MDA and the non-canonical increase. To me, it would seem like they're both increases in AMPA receptors at a specific synapse, and they should have sort of similar results, but they give you such distinct behaviors.
Yeah, thanks for the question. So, I mean, the increased AMPA-NMD ratio is really a proxy of something that has changed and doesn't tell us whether it's the AMPA that increased or NMDA that is decreased. And this is something that we are currently looking to that. So, which is the real mechanism, synaptic mechanism underlying this change in AMPA-NMD ratio, right? So, the ratio can be one or the other. The change in GLUE2 lacking AMPA receptor, presence of GLUE2 AMPA receptor is dramatically different from baseline because this receptor flux calcium at resting membrane potential. So it means then people have shown that when these receptors are present, there is a change in Hebbian plasticity. So there is the anti-Hebbian form of plasticity because you are able to stimulate synapses at resting membrane potential because you don't need even the NMDA, the calcium from the NMDA. So dramatically at the level of the synapses, there is a big difference in signaling whether you have GLUE2 lacking AMPA receptor with non-canonical AMPA receptor or you don't have them. That is very different to a synapses where you just increase the number of AMPA receptor without changing their quality.
In terms of calcium
Yeah.
entry.
Rob, go ahead.
Yeah.
Thanks, that was great. The 1 versus 10 hertz effects are also really interesting. Why do you think 1 hertz induced LTP and 10 hertz didn't? It's a little bit different than the usual rate-based rules for synaptic plasticity, right?
So I have not, sorry, I did not.
Oh, you didn't hear? Okay.
No, yeah, I hear very badly here, yeah.
Why did 1 hertz induce LTP, but 10 hertz did not? It's a little bit different than the usual rate-based forms of LTP for glutamatergic synapses.
But this is the thing, so I have never used the word LTD or LTP expressly because we do not know yet whether this is the change in AMPA and MDA ratio is a decrease of an MDA or increase in AMPA. And so 1 hertz generally in certain synapses generate LTD-like, so a decrease in AMPA receptor. We are doing that type of experiment on the slides first, and then we will look. I mean, you have also to consider that from the moment where there is the activity of these neurons in vivo and the moment that we perform the patch clamp, it passes 24 hours. So it's not necessary that what is happening at the time 0 is how is expressed then 24 hours later. So this is the type of work that we are trying to look into sliced work.
Great. Let's thank Camilla.
Thank you.
All right, so while we get set up, we'll welcome Caroline Jha.
Is that good? Maybe we'll get a connector.
Technical challenges. Good. All right, here we go.
Hi everyone, my name is Caroline Jia. I'm an MD PhD graduate student working with KTI at the Salk Institute. Today I'd really like to thank the organizers for giving me this opportunity to tell you a little bit about my thesis work entitled Social Exclusion Changes the Neurorepresentation of Physical Pain Within the Anterior Insular Cortex. How many of you over the past few years have felt socially isolated or excluded, maybe even rejected or jealous, or maybe even lost a loved one? Would you describe these social experiences as being painful? As a social species, losing our social ties can be one of the most devastating and painful experiences that we can go through. But is this emotional pain similar to what we think of when we think of physical pain, when there's actual or potential damage to our bodies? Interestingly enough, within the English language, physical and social pain are similarly conceptualized, such that the same words are used to describe both physical and social injury, such as broken bones and broken heart. And this is also seen in multiple other languages, suggesting that this overlap between social and physical pain is a universal human experience. The pain overlap theory proposes that the experience of social and physical pain overlap by potentially sharing some of the same underlying processing systems within the brain. And this has been shown in various fMRI studies of social exclusion, where regions such as the anterior insular cortex and the anterior cingulate cortex have been implicated. However, we don't know the specific neural substrates for where these two types of pain can overlap in circuitry. And in general, how is this abstract experience, such as social pain, even represented within the brain? And where and how can it modulate sensory pain? So to address this question, we designed a novel social exclusion task, which we call the FOMO task, after fear of missing out. And then using this paradigm, we were able to record from the cortex and really delve into how social and physical pain are represented within the brain and how these two types of pain may potentially overlap. So within this paradigm, mice first undergo individual associative learning. And then on test day, one mouse is excluded by a barrier and observes its cage mates on the other side collectively consuming a chocolate milk reward. And we can see on the left-hand side this purple dot, which we would call FOMO behavior. And this is really when the mouse is actively rearing or climbing or orienting towards the other side. And so how do we choose these specific behaviors that we define to be FOMO behaviors? Well, to do so, we use sleep first to perform pose estimation on this excluded mouse and extracted various features of the mouse, such as its orientation and size, as well as its velocity and angle to port, distance to port, and angle to itself, and tortuosity, which is the path that it takes within the chamber. And using these specific clusters and features, we were able to do unsupervised clustering and extract different behavioral motifs, which we can see examples of in this video. So within this unsupervised clustering induced map, we can see various different behavioral motifs. And we can classify them into two main categories of either being FOMO behaviors, where the mice for active FOMO are actively rearing or climbing to get to the other side. There's also passive FOMO behaviors where the mice are passively orienting or passively looking to the other side. And we also have fine behaviors in gray where the mice are not oriented to the other side. So there are a couple of very interesting features within this paradigm. So in order to account for these features, we counterbalanced with a couple of controls. So to control for being in the chamber, hearing the tones, having an inaccessible reward, we have the socially neutral tone-only control. And then to control for being in the chamber, hearing the tones, having this inaccessible reward, and visualizing another mouse consuming this reward, we have the one mouse control. And we think this one mouse control can also be a type of social pain, similar maybe to the experience of envy or jealousy, where you want something that someone else has. So then if we take these 60 trials within each of these paradigms, and then we separate them to being either predominantly filled with FOMO behavior or fine behavior, we can see that mice undergoing social exclusion spend a significantly greater amount of trials engaging in this active FOMO behavior, suggesting that they're really making a stronger effort to be reunited with the other side, which would alleviate their emotional pain. And then we wanted to assess if there's any changes to their physical pain behavior after social exclusion. And so to do so, we placed the mice on a hot plate and recorded the amount of time that the mice spend attending to their wounds. And we can see that there's actually a negative correlation with the amount of FOMO trials that the mouse has and licking behavior, which we don't see in any of the other controls. And this really suggests to us that the more excluded behavior a mouse exhibits, the less it will attend to its wounds. And taking a step back, this is really interesting because one theory of why animals even exhibit pain behavior to begin with is to draw attention to themselves from their social group for help. But perhaps after this aversive social experience, there's less of a drive to do so. So now that we've characterized this novel task and shown its effect on modulating pain, we then wanted to see how these two types of pain are represented within the brain. So we have a couple of models for how these type of pain can potentially overlap. So the first model is the spatial convergence model, where potentially physical and social pain activate the same pain responsive ensembles. Alternatively, we have the temporal convergence model, where potentially social pain induces the release of some neuromodulators, and this gradual accumulation of neuromodulatory signal will lead and recruit more neurons to the pain responsive ensemble. And so in order to test these models, we recorded using cellular resolution imaging within the anterior insert cortex and recorded specifically during the social paradigm, as well as during nociceptive and non-nociceptive stimuli that we applied afterwards. And then we took the neural activity that we obtained from these trials and plotted neural trajectories. And what we can really see here is that the subspaces for each of the paradigms is very distinct. And we can see that the trajectories between FOMO and fine conditions are also very separable. So we wanted to really test the separation and see if the paradigm and the different behavioral trial types can be differentiated using decoding analyses. So in order to do so, we trained a generalized linear model, GLM, with FOMO and fine trial neural data. And we can see that we can strongly decode between these FOMO and fine trials within the AIC. And this is really intuitive because these two different types of behaviors are very, very different. So then we wanted to test if we could decode between the three paradigms. And we can see that, again, it is possible to decode between all three of these paradigms, which really suggests that the AIC is able to encode these different social experiences differently. So then we wanted to assess any changes to the neural representation of physical pain after this experience. And so in order to do so, we again trained a GLM with various neural activity in response to stimuli. And we can see that after the social stress that we induce using social exclusion or one mouse, there is an increase in the decodability of different physical pain stimuli when we compare it to tone only. And this really suggests potentially a mechanism for enhanced physical pain after social pain. So now that we've demonstrated that social and physical pain can even be represented within the AIC, we then wanted to see how these two types of pain could converge. And so, in order to test Model 1 for spatial convergence, we co-registered the neurons across these two different pain states. And we can see on the left-hand side that the neurons that respond to either the FOMO trials or the FIND trials do not significantly overlap with the neurons that respond to pinprick, which we would think would be extremely aversive. And so this suggests to us that potentially Model 1 might not be a mechanism for the overlap between these types of pain. So then we turned to Model 2 for temporal convergence, and we tested various neuromodulators such as dopamine and oxytocin, but we didn't see any meaningful signaling dynamics during our task. And so then we transitioned to looking at the endocannabinoid system, because the endocannabinoid system has been shown to be able to suppress nociceptive processing. And so we used the endocannabinoid sensor from Yulong Li's lab and recorded during both the social task and during the physical pain stimuli. And then we can see specifically during the social task, there's an increase in the endocannabinoid dynamics, specifically only during the FIND trials, but not during the FOMO trials. And we don't see this occurring in any of the other controls, suggesting that it is specific to being excluded from the entire group. And we think that potentially these endocannabinoid dynamics might be playing a protective role such that the mice don't need to engage in this active FOMO behavior. And then if we look at different nociceptive and non-nociceptive stimuli afterwards, we can see that specifically after tone only, there's an increase in these dynamics, suggesting potentially that this is a mechanism to suppress the nociceptive processing. But after one mouse or social exclusion, this effect is abolished. And this could be potentially also a mechanism for increased pain, which really suggests to us that the endocannabinoid dynamics could play a protective role for both social and physical pain and could be a neuromodulatory mechanism to bind these types of pain. So overall, we've demonstrated that social exclusion can modulate physical pain and alter and enhance pain stimuli decoding, and that endocannabinoids could be a neural substrate that really support this temporal convergence model for the pain overlap theory. And none of this work in general could have been done without my mentor, Kei, as well as other collaborators and members of the Tai Lab. And I can honestly say that the Tai Lab is such an incredible training environment, and I highly encourage anyone interested to apply because we are recruiting. So thank you so much for your time and attention, and I'm happy to take any questions.
Thanks for a very cool talk. We have time for a couple of questions. One right here.
Hi, thanks for a great talk. I was wondering if you try to look at some autonomic measures of pain.
Yeah, that's a really great question. Thank you so much for asking it. I do think that that would be a very interesting thing to look at to see if there's also other affective measurements that could be effective, such as anxiety with like increased heart rate or blood pressure. I think that would be really interesting to look at, but we haven't done so yet.
Yeah, I would like to challenge the concept that you're presenting. I don't think looking at the fMRI signal for pain representation in a region can be interpreted as being, back translated into some concept of sharing different types of aversive states as being the same. A nociceptive response is necessary to show a pain perception that you can link to a somatic representation. I don't think you showed that in this data.
Oh, sorry. Could you repeat your question? Stand a little closer. Sorry.
I'm sorry. I think I'm making a statement in a sense that I think there's a confusion of categories. Just because we use the language of pain for my heart and for my foot, my injury-related representation and my affective states of not liking something should not be confused with each other. And in a sense, your data shows that even in the anterior insular cortex, these representations are in unique neuronal populations. So your alternative explanation for your data would be that they do not overlap.
No, yeah, that's a really great point. We're definitely thinking about whether or not social exclusion modulates the affective component of pain, which is the emotional component, like you mentioned, or the sensory component. And we are currently doing more analyses with the sensory component, such as analyzing the data specifically on the hot plate. And we do see a trend towards increased sensory pain as well. And so we really think that potentially this paradigm is affecting both of the components of pain, but I do agree that that's a very clear distinction that we do have to make specifically when we're thinking about pain behavior, since it's so complex with both the changes in nociceptive threshold as well as changes in the emotional affective state of the animal. Yeah, that's a very good point.
Great, thanks. Let's thank Caroline and keep going. And while this connects, I'd like to welcome our next speaker, Aishwarya Balwani from Georgia Tech.
Awesome. All right. Thank you so much for the opportunity. I'm Aish, and I'll be talking about some of the work we've been doing towards trying to understand what the architectural biases are in the canonical cortical microcircuit. So there's a long-standing belief that the brain, and the cortex in particular, is an hierarchical and predictive operating system of sorts. And when we talk about this, what we are typically speaking about is the brain's ability to be able to compare top-down predictions with bottom-up sensory signals and use this information towards learning and inference. And what this automatically implies very often is the ability to distinguish between expected and unexpected signals is quite pivotal to learning effectively. And there's been a lot of experimental evidence corroborating this hypothesis, so to speak. For example, when mice are trained to learn a sequence of Gabor images, introducing a surprise image in the sequence typically invokes excessive activity in cells. Likewise, the presentation of, say, surprise versus unexpected stimuli tends to invoke, again, varied responses in deep versus shallow neurons in different layers of the cortex. And finally, even with just natural images, if you have a sequence and the mice learns this sequence, you see that over time, familiar images show a reduced response, whereas taking out an image that it is expecting to see, again, increases the response in certain cells. And of course, there's been work done towards trying to marry both all of these ideas and have a nice idealized theoretical framework for it. And the two components of this in the computational sense is the very famous predictive coding framework. And in the structural sense, what people often like to think about is the canonical cortical microcircuit. And again, the idea with both of these is that lower areas, which sort of correspond to the feed-forward circuit in the microcircuit, encode errors and sensory information, while the higher areas, which now corresponds to the feedback circuit, typically deal with predictions of what's happening in the lower areas themselves. However, despite all the work and all the theorizing, there's still a lack of understanding and a comprehensive sense for exactly how the structure in the cortex or the cortical microcircuit actually affects neural representations, their geometry, and their computational roles. And so towards that end, the goals of this project are threefold. For one, we want to see exactly how the information is encoded in the microcircuit, if there are any inherent benefits to this architecture, because we see this across mammals of various kinds. And finally, if the neural representations are shaped by the architecture or if they're more objective dependent. And we choose to try studying all of these questions in a computational sense, with a set of in silico experiments and using recurrent neural networks as a model. So to start, this is what we're calling the canonical cortical microcircuit. We have a lower and a higher cortical area, and each of them is essentially a simplified version of the cortical column. We use RNNs to model the dynamics of the different layers, so layers 2, 3, 4, and 5, 6. And we make sure that the ratio of neurons in each of these RNNs is consistent with what you'd see in the mouse visual cortex. The interareal connections, which are essentially the green and the orange arrows that you see, have a sparse and fixed backbone that we choose at initialization, but its weights are learned, and the backbone itself is sampled from a Bernoulli distribution. And we make sure that all these projections are strictly excitatory, since long-range projections in the brain tend to be so. And we also make sure to incorporate time delays, which you normally see. So there is a one-step time delay in the feed-forward signal that is being projected using the green arrow, and a two-step time delay in the feedback signal that you receive from the higher to the lower cortical area. And finally, we learn all these weights using backpropagation through time. The task that we're dealing with, or the one that I'm going to go into some detail now, is a very simple sequence memorization task. You have a set of sequences where the first three elements are permutations of each other, and the last element sort of is an identifier for that particular sequence. And the loss function that we use to train the network is your standard mean squared error reconstruction loss, where you're now training the network to be able to predict B when it sees A, C when it sees B, so on and so forth. After training the network on a clean set of sequences, we also subject it to two kinds of perturbations or violations. The first is a temporal violation, where the network now sees ABB instead of the expected ABC. And the second is a spatial violation, where we add Gaussian noise to the overall representations, where the noise is sampled from a Gaussian distribution that is zero mean and of different amounts of variance. So the first question that we try asking or answering is, where exactly are task-relevant variables encoded in the circuit? And by that, what we're trying to see is where in the circuit they're most linearly decodable. So if you've not seen this figure 1,000 times like I have, there is a legend on the top right, which will show you exactly which RNNs I'm speaking about. There's a color correspondence. And so when we're speaking about lower level information, so in this case, it is just what input state or what image my microcircuit is receiving. What we find is the lower areas, so the blue and the orange ones, tend to be most decodable. The way you want to sort of interpret these figures is pay attention to where the most weight is in that distribution. A slightly more abstract concept, which is where in the sequence am I? So am I seeing A, which is in position 1, versus am I seeing A in position 2, is better encoded in the higher cortical area, and in particular, higher layers 2, 3, and 5, 6. And perhaps what is most cool or most exciting is that the ability to decode surprise or unexpectedness when you use some degree of temporal violation in your sequence is best encoded in the layers that actually receive the feedback, which are the lower layers, layer 2, 3, and layer 5, 6. We also looked at the geometry, or in particular, the dimensionality gain of our representations. And what I mean by dimensionality gain is just the ratio of my linear, which is global dimensionality, to that of my nonlinear or local dimensionality. And the way you want to interpret this is that if you have a high dimensionality gain, such as in this case, you're typically able to represent a certain degree of complex yet structured and smoothly varying stimuli. Whereas in the case of a lower dimensionality gain, your linear and nonlinear dimensionalities are practically the same, and therefore it is highly likely that the stimuli that you're representing in that particular case is rather unstructured, almost definitely very nonlinear, and potentially very noisy. And so if you look at what's happening over here, we see that the dashed dimensionalities or dimensionality gains are those which belong to the higher area, and they tend to be significantly, or at least somewhat clearly, above those of the lower cortical area. And what this sort of tells us is that because the overall dimensionality gain of the higher area is greater than that of the lower ones, the higher ones are encoding some nonlinear but still structured information. More simplistic, linearly decodable information is being encoded in the lower area, and this generally agrees with the decodability results that we just saw. We then try understanding better how exactly the inter-areal feedback connections actually affect the representation across the microcircuit. And so to do this, we go through a certain number of strategically motivated structural alterations. So this is the canonical cortical microcircuit, or the cortical RNN that we've been seeing so far. We see a no feedback architecture where we just get rid of any feedback, inter-areal feedback that's happening, but the feed-forward projection still exists. We also check a bidirectional feedback architecture where we now have or use the same channel to do both feed-forward and feedback projections, but with a time delay. We also check a unidirectional feedback architecture where we don't project back to the area which projects forward, but we still keep the other one. And finally, we do a same populations control where the skeleton or the structure is exactly the same as the cortical RNN, but now instead all the number of neurons and all the RNNs are the same. So there's no natural projection or expansion or compression of signal that's happening. So when you check what the results are in the presence of either spatial or temporal noise, what you find is that just the architecture in itself does not make much of a difference to the overall performance of the different architectures. However, what is very cool is that if you try understanding which layers are able to decode surprise or unexpectedness, you find that layers which receive feedback, which are represented in the green bars in this case, do a much better job as compared to those which don't get feedback, and the other ones just sort of perform at chance or like a little higher or lower. And so what this sort of tells us is that interareal feedback is really, really important for us to be able to decode expected versus unexpectedness just at the get-go. We also tried a couple of other tasks. I'm not going to go into too much detail regarding these, but just the greatest hits. We tried a spatial traversal task where now the microcircuit receives an input state and also an action and has to, in a sequential manner, figure out where it's going from point to point. And what we see is that, again, the input state very nicely decodable in lower area 4. The position is nicely decodable in an area which gets feedback, that's the lower 5-6, and a higher area, which is higher area L5-6. Surprise is best encoded in lower area layer 5-6, and the overall dimensionality gain is greater for the higher area versus the lower one. We also do a simple rule learning task where, given a binary representation, the circuit now needs to predict a binary representation for x plus 1, and so then you do x plus 1, x plus 2, so on. And again, very similar corroborating results where input states are best decodable in lower area L4, the output state or the next state is most decodable in the higher areas L2, 3. Surprise, again, very nicely encoded in lower area layer 5-6, and higher area dimensionality gain is greater than that of the lower one. And finally, a third question that we try answering is how exactly does changing your loss, and in particular including a sense of predictive error, actually affect information representation across the circuit? And so to this end, we train the network slightly differently and use a two-phase training scheme that is inspired by Rao and Ballard's work. And so what we now do is use the same reconstruction loss to train the parameters in the lower cortical area and the feed-forward projection, but use a predictive loss, which essentially is the mean squared error in activity between what's happening in the higher areas L5-6 versus the lower areas L2, 3 and L5-6. And we do an alternate training scheme, and we train across both phases for an equal number of epochs. And so intuitively, what's happening now is that we're trying to encourage the higher areas, layer 5-6, to predict the next step predictions being made by the lower areas, layers 2, 3, and 5-6. So for the simple tasks that I sort of showed you, again, what we found is that there's no real significant change in performance across the tasks or architectures. However, and this is sort of a fun hypothesis that we've got, is that the addition of the predictive error inherently encourages the layer receiving feedback, in this case L5-6 in the lower area, to specifically encode unexpectedness, which goes along incredibly well with what predictive coding theory postulates. And so the evidence towards this end really is what we see when we look at the dimensionality gain of the different RNNs in this particular case. So what you see is that the green line, which corresponds to the lower areas L5-6, is really decreasing in the cortical RNN, whereas it's about the same as the rest of them in the no feedback architecture, where it does not receive any feedback. What this decreasing or lower DGs pretty much implies is that it's encoding some degree of unstructured, non-smooth aspects of your stimuli. And this is a bit of a leap of faith, but we say that it could be noise or surprise. And it's not just with those two, but what we also see is that the same thing happens across the different architectures, where the middle two that are in the box, you see that the green line does not really show any significant difference from the other ones, but when you compare it to the other architectures which do receive feedback in that area, you have this nice decreasing trend. And again, you have same corroborating results with decodability, where if you see towards the very end, all the weight in the green area is very nicely centered around 1, showing that surprise is encoded almost perfectly over there. So in conclusion, we find that there does seem to exist a natural functional modularization of representations in the cortical microcircuit, and that as constructed and trained, the microcircuit does seem to have a natural inductive bias for separating simple stimuli in sequential tasks. And finally, that using predictive error, while just right now for these simple tasks does not do much, it does help the microcircuit encode unexpectedness and surprise significantly better. And to finally finish off, a big thank you to the people who make this work possible. Suhi, who's been a great friend and helped with the experiments, Hannah, who's advised this entire project, the Math Neuro group at Georgia Tech, who are my lab mates and are incredibly supportive, and the NIH for their generous funding. With that, I'd like to finish off and open up to any questions.
Thanks very much. I think we have time for maybe one question.
Oh, yeah, I have a question. Very nice talk. So I was curious, predictive coding theories kind of differ in whether you have errors kind of explicitly coded in firing rates of neurons versus just more implicitly coded in the network activity. Did you see like a rising of more like just error neurons, or was it really just more in the decodability of the activity?
I don't think we saw it, the latter definitely. Mm-hmm. I don't think we saw anything towards the former, but I also didn't like explicitly check it in too much detail. But yeah, it's just, also we're not really encoding it explicitly. It's more like implicitly encouraged. So that might be it, but yeah.
Okay, thank you.
Sure, thank you.
Great, thanks very much, Aish.
Thank you.
Great, and so while we plug in, we'd like to welcome our last speaker of this session, Haleigh Mulholland. Welcome.
There we go. All right. I'd like to thank the organizers for giving me an opportunity to talk about my research today, which is looking into the mechanisms that contribute to the self-organization of pattern activity in the developing visual cortex. So a hallmark of the visual cortex of primates and carnivores is the columnar modular organization of cortical activity. What I mean by modular in this case is that local cells tend to be coherently responsive with their neighbors to similar stimuli, such as oriented bars of light. And they are functionally connected with distance populations of neurons that respond to similar stimuli, forming a distributed network of correlated activity, which also produces the beautiful canonical orientation preference maps that we see in V1. And the circuit mechanisms that contribute to the development of these networks is a key question in theoretical neuroscience. And there is decades worth of computational modeling that tries to describe how modular activity could emerge in the cortex. And one of the key underlying assumptions of a lot of these models is that this modular activity arises through interactions within a network that it comprises of local excitation and lateral inhibition. And this mechanism was actually first described by Alan Turing as a way to model how autonomous pattern formation could occur in biological systems. And it's extremely useful in a neuroscience context because it allows networks to be able to structure unstructured inputs, let that activity iterate through a network of short-range interactions to produce highly structured modular activity patterns. And these activity patterns are very periodic and have a very specific wavelength, which is set by the relative extent of the excitation and the inhibition to one another. And despite the fact that these ideas have been around for a long time and how useful they are, a lot of key predictions about this particular mechanism has not ever been tested in vivo. And so today I will show you work trying to find evidence of something like a Turing mechanism in the visual cortex. The context that we're going to be searching for this mechanism is actually going to be very early in development. So in ferret primary visual cortex, before eye-opening, there is spontaneous activity patterns that are highly structured and modular, which reveal large-scale networks of correlated activity that predict the eventual orientation preference maps found in these animals at eye-opening. And we see long-range correlations activity at a period of development before there is any direct horizontal connections with long-range axonal connections between these regions. And these modular patterns persist when you block instructive feed-forward input into these networks, suggesting that these neurons are able to coordinate their activity using a dynamic self-organizing network. So in order to test the extent to which these early cortical networks can self-organize activity, we designed a custom microscope that allows us to do simultaneous optogenetics and wide-field calcium imaging. And so we can create arbitrary stimulus patterns, shine those patterns onto the surface of the cortex, and selectively stimulate populations of neurons with spatial specificity on a scale that reaches millimeters. And the framework that we are doing these experiments will be to virally express GCAMP and redshifted opsin crimson in excitatory cells in ferret primary visual cortex layer 2-3. And we will be doing all of our calcium imaging in this period of time prior to eye opening. And we will collect both baseline spontaneous activity and also responses to optogenetic stimulation. So if early cortical activity organizes itself using a Turing mechanism, we can make some very clear predictions about how injecting activity into these networks will produce outputs. So the very first prediction is this idea I've already introduced, which is that a uniform or unstructured input into these networks should produce modular output activity. And because these networks produce this modularity by amplifying initial noise fluctuations at a very specific, in this case, critical frequency, or otherwise I'll be referring to it as the intrinsic wavelength of the network. What this means is that different noise inputs into these networks can produce different modular patterns. So to do this experiment, we have the animal sitting in the dark. Its eyes are shielded from any other sources of light. And we are using a uniform full field of view optogenic stimulus that covers the entire cranial window. And when we stimulate with this stimulus, we see the rapid emergence of a strong, robust calcium response, which elicits a modular pattern. So you can see domains of activity rise up, such as here, and they're coordinated with other populations of neurons that extend over millimeters of the cortex. And repeated stimulations with the same exact input is able to elicit different output patterns. And when we quantify the wavelength of this opto-evoked activity, we see that it very regularly produces activity with a wavelength of about 0.8 millimeters between active regions, which is consistent not only with the spacing that we see with visually evoked responses, but also with the spontaneous activity that occurs without any stimuli. And as I mentioned, repeated stimulation with the same flat pattern is able to elicit a diverse array of activity patterns. We can see that variation in the trial-to-trial event correlations across all of our repetitions. And we attempted to quantify this variation by doing principal component analysis and saw that the dimensionality of our data set is sitting around 10 to 12 linear dimensions, which, as you'll notice, is higher than 1, which is what we were putting into the network. And in a series of experiments I don't have time to share with you today, we also demonstrate that these modular patterns emerge when you block feed-forward input into the network, and that these modular patterns require activity propagating through intercortical circuits in order to become modular, which altogether shows that these variable modular patterns are likely emerging out of intracortical interactions. So we've established that these networks are self-organizing. The next thing we can test is this idea that if the noise that is amplified within these networks is what's responsible for producing these different activity patterns, then we should be able to influence the activity if we impose a structured input into the network. And specifically, if that activity pattern is consistent with the intrinsic wavelength of the network, then that specific pattern should be amplified, meaning that the input should be pretty similar to the output. So to test this, we designed some structured optogenic simulation patterns by bandpass filtering white noise. And the wavelength of these patterns is consistent with the wavelength of endogenous activity in the cortex. And we alternate between stimulating with these three patterns to see how specific are these cortical responses. And we find that this structured optogenic activation of cortical activity produces evoked responses that are similar to the stimulus pattern. We see that on both the individual trial-to-trial event level and also robustly in the mean response across trials. And if you ask how similar are the trial-to-trial responses, events that were driven by the same stimulus pattern tend to be selectively and strongly correlated with one another. And if you also ask how similar is this evoked activity to the actual pattern of the stimulus, we can see that we see significant correlations in these two patterns. All of this is to suggest that our ability to directly influence the activity pattern based on our artificial inputs speaks to these networks' ability to self-organize based on inputs into these networks. So we've established we can influence the activity patterns. The next question, and the final question we ask is, well, what happens when you try to drive these networks with activity patterns that don't match the endogenous spacing or the intrinsic wavelength of the network? What we'd expect is, if these networks work by selectively amplifying activity at their intrinsic wavelength, the input pattern should become biased towards the intrinsic wavelength of the network. And so we can see this in a linear rate model with local excitation and lateral inhibition interactions. We can drive these models with structured input patterns of varying spatial wavelength, and then ask how does the input compare to the output. And what we see is in these networks, the individual events tend to be very consistent in their spacing, and that spacing pretty much matches exactly with the intrinsic wavelength of the network, which we see here. And we can see direct evidence of this network transform when you look at the mean activity across simulations, which shows this bias, again, away from the input wavelength and towards the intrinsic wavelength of the network, but it's more of a compromise, so it resides on a slope that is between the two values. We do similar idea in vivo, where we are designing, again, these random bandpass filtered patterns, but now we are varying the spatial wavelength. And we can see that driving with these patterns produces individual events that tend to have a very consistent spacing, and that the spacing is consistent with the intrinsic wavelength, essentially regardless of the optogenic input pattern. And when you look at the average response across all trials, we can see actually the structure of the mean response is affected, where you see this in the smaller wavelength stimulus, you also see a smaller wavelength mean response pattern, but this mean response pattern is a compromise between the input wavelength and the intrinsic wavelength of the network. These results demonstrate that regardless of the input structure, these early cortical networks are strongly biased to produce activity at its intrinsic wavelength. So we've demonstrated that these cortical networks can self-organize uniform inputs into modular output patterns, that structured inputs can selectively influence the activity that we're able to evoke, and that these networks are strongly biased to produce activity at a very specific wavelength. And all of these experiments strongly support that these early cortical networks self-organize their activity according to a Turing mechanism. And to our knowledge, this is the first demonstration of this principle examining neural activity in vivo, and it supports decades worth of computational modeling trying to describe how these distributed networks emerge in development. And I will end with one final thought, which is to return back to this idea of is this actually the mechanism that is responsible for producing endogenous activity patterns? So we've been shining all sorts of artificial inputs into these networks. How similar is our responses to the activity that the network can naturally make? So returning back to our uniform object simulation experiments, we compare the opto-evoked activity to the spontaneous activity and see that for the majority of our opto-evoked events, we can find matching activity patterns that occur spontaneously, and that the strength of the correlations are stronger than would be expected by chance. And as I also mentioned earlier, the wavelength of spontaneous activity is very similar to the wavelength of opto-evoked activity, suggesting that these two data sets are constrained by a similar network mechanism. And since, which altogether would support that these early spontaneous patterns are emerging from intercortical interactions through a self-organizing network that is using a Turing-like mechanism. And since these early spontaneous activity patterns predict the future functional organization of the network, it shows and really highlights the importance of this mechanism in establishing the network's use for visual perception. I'd like to thank both my advisor, Gordon Smith, and also close collaborator, Matthias Kaschube, for their help with this work, and also the Smith and Kaschube Labs for their support and feedback. And I'd like to open it up to any comments and questions.
Great. Thank you very much. So we do have time for a couple of questions. So one here.
Very interesting work. Thank you. Do you think that you could change the characteristic wavelength by doing something like modulating inhibitory activity? And do you think that if you could do this chronically through, say, administering some, say, like a GABAergic compound, that you could cause long-term developmental changes in the characteristic scale of adult ferret visual cortex organization?
So that's a great question, is like, what can we do to try to change the spacing of these networks? Is there anything that we can do to change inhibition specifically? So previous work has done pharmacological implications of the idea. So diazepam, I believe, would lead to an increase in the spacing of these ocular dominance columns, which is a related idea to orientation selectivity. And I would imagine that if you could do this in a more controlled way, maybe with all optical approach and longitudinally over time, you might be able to see some sort of plasticity like you imagine. But I think the specific experiment would be, it still has not been done yet.
I see there are a number of people lined up for questions, but we are out of time for this session. So I strongly urge you all to come up and chat over coffee. But thank you, Haleigh, and can we give one more round of applause for the whole session? Thank you all.
interesting work about oxytocin, I believe. You're getting the sympathy.
Yeah. Oh boy.
So I'm going to hit you at 30 minutes. Maybe switch screen.
Where's the?
There's a switch screen. I don't use PowerPoint, but I know there is a switch screen button, right? If you go to the slideshow, might there be a? What if you just take that? Ah, there we go. No, that's not the one. The other one. And try one more.
Oh, great.
There we go.
Okay. Hi, everybody. Thank you for your patience. I'm going to skip over some stuff in the interest of time. What I'm going to be talking to you about today is the work in our lab on mouse maternal care and oxytocin. I'll start by going over some background information, some published work on oxytocin and a form of mouse maternal behavior called pup retrieval. Then I'll be really focusing on the responses of oxytocin neurons to co-parenting, and I'll end the talk with some unpublished data on the challenges of single parenting, and really just how difficult it is for mothers to keep their babies alive. The people shown here on the slide are the ones who've done all the work I'll be talking about today, current and former graduate students and postdocs. Bianca Marlin, Ioana Carcea, and Silvana Valcheva now have their own labs at Columbia, Rutgers, and Cologne. For those of you who are graduate students, if you're thinking about doing postdocs, please consider their labs. They are fantastic mentors and amazing scientists. So the way we got into oxytocin in the first place, my lab historically has worked on synaptic plasticity, and more or less we were interested in examining some behaviors and some stimuli that had interesting properties. I think we had a lot of problems with AV, so let's see if this works. You can imagine a baby crying and just how, or laughing, and just how important and meaningful those cues are to parents. For example, in this case, the baby's crying and the mother tries to feed the child, but it doesn't seem to calm the baby down. The infant keeps crying. Something else is the matter. So as parents, we have to quickly figure out what these cues mean, right? The child can't say it's hungry, it can't say it's tired. It can really only just cry. And we have to rapidly and basically perfectly figure out, I mean, that's the only sound, so.
All right, that was terrible.
Okay, sorry.
I'm going for worst host over here.
Oh no. Oh, plug this. Oh no, I don't need the sound. Oh, okay, great. Nope, no sound. I'm good. Okay. Thank you for all appreciating this. So it's not just human moms and dads have to figure out what baby cues mean. Animal moms and dads have to do this too. And if there's one thing we've learned about the biology of parenting, it's that the neurohormone oxytocin is absolutely critical. Oxytocin is a 9-amino acid peptide. It's made largely in the brain in a hypothalamic nucleus called the paraventricular nucleus, also the supraoptic nucleus. It's not a trust drug, it's not a love hormone, it's just a neuropeptide. There's really been a huge revolution in terms of our understanding of the oxytocin system in the last decade or so, really helped along by mouse genetics. Most oxytocin is synthesized and released by the posterior pituitary for the two critical actions of oxytocin for maternal physiology: milk ejection for nursing and uterine contractions for parturition. But really over the last decade, there's been a lot of work understanding the projections into the brain. And we've been wondering what those projections do, how they're active, and how they modulate and maybe enable plasticity for parental care. So one of the important forms of parental care that mice express is called pup retrieval. Here's a cartoon. I'll show you a movie in the next slide. Basically, mice are prey species, right? They have to move their nests around in the wild to escape predation. The pups are born blind and deaf. They really don't do much for the first week or so of life. But the mother is constantly kind of going in and out of the nest. Every now and then a pup also gets dragged with her if it was attached onto her. And if the pups are then isolated from the caregiver that provides them with warmth, they start changing the ultrasonic vocalizations they're making. They shift up to the high ultrasound spectrum. They're emitted at a characteristic bout rate of around 5 hertz. And the mother animal uses this cue to go find the pup and take it back to the nest. We can study this in the lab, just like this. Here's the home cage of this maternal animal in the back. The pup is calling. We use ultrasonic microphones to verify it's making sounds. Watch what the mother does. Tend to her behavior. She understands how heavy the pup is, knows exactly where to pick it up to overcome gravity without applying too much pain. And so there've got to be motor aspects of learning here in terms of the mother understanding how to appropriately take care of the pup. And really a lot of the work in our lab is about sort of the interplay between innate and learned aspects of these behaviors. Because certainly virgin females don't act like this. Here's a virgin. She might go and investigate the pup as a source of novel sound generation, but she certainly isn't fetching it like the mom did. And so when we got started on this, way back when Bianca joined the lab, we wondered how might oxytocin interact with the experience of becoming a parent for the first time. And so initially, to kind of factor out all the complex issues related with pregnancy, we focused on alloparenting, taking naive, nulliparous virgin females, co-housing them with an experienced mother and her babies. We knew in the field that the virgin will eventually become a co-parent, and we asked would oxytocin speed this up in some way. So in some cases, we just inject the animal with oxytocin. In other cases, we infuse it into the brain. In other cases, we use optogenics and stimulate release of endogenous oxytocin compared to, for example, saline injected control animals. And here are just 3 animals. These are virgin females. They're co-housed with the mom starting at time 0. The retrieval success of the virgin is on the y-axis. We test her just like I showed you in the previous slide. You can see the animal given saline takes about 2 days before she starts retrieving. The behavior goes from completely off, 0% retrieval, to 100% retrieval, perfect success. And oxytocin, either endogenous or pharmacological, speeds that up by about a day, day and a half or so. And then other work in the lab by Jen Schiavo sort of did the opposite, or a loss of function experiment, asking, okay, so if oxytocin supplements enhance alloparenting onset in virgins, what about suppressing activity? And so she expressed halorhodopsin in the oxytocin neurons, suppressing activity of the system when animals were exposed to pup cues, and those animals retrieved less often and less successfully, as shown in the plot on the right. Now, a lot of the work in my lab is focused on cortical representations of pup call sounds and how it is that cortical neurons maybe change with experience and generate a pup call category, specifically lateralized to left auditory cortex. I'm not going to talk too much about that work, except to show you some new data from a postdoc in the lab, Amy Lemessurier, who is asking, using longitudinal 2-photon imaging from certain layer 5 projection neurons, what neurons develop sensitivity to pup call sounds. We had a hypothesis that given how kind of stereotyped and reliable the behavior was, that maybe it was the cortical striatal neurons that were most important for this. But actually it turns out that the cortical striatal neurons don't really have pup call responses themselves. That's shown here. On the other hand, cortical fugal back projections to midbrain really did develop nice responses, and both categories have responses to pure tones. She also verified that it was the cortical fugal projections to midbrain that became pup call responsive using optically tagged wholesale recordings in vivo, using retroviruses to express channel rhodopsin, putting a fiber optic and a patch pipette, patching onto cells, and showing nice responses to pup calls after experience in these cortical fugal projections. But for the rest of my talk, I really want to focus not on the cortex, but on the oxytocin neurons themselves. This experiment that we did indicates that the oxytocin system, even in a virgin female, must be online to help shape her behavior in some way. She's not lactating, so what kinds of experiences with the pups or with the mother, what kinds of experiences might be activating her oxytocin system when she's co-housed? So to look at that, we've basically been doing 24/7 documentary filmmaking of the home cage life of these mice, putting cameras usually on the top, now sometimes on the side or underneath, and recording initially for days, now for weeks to months, continuously, just to ask about the spontaneous behaviors these animals emit when they're living together, being parents. We also combined this with phototagged recordings from oxytocin neurons in the virgin female. The PVN, this part of the hypothalamus, is deep, it's midline, it's mixed population of cells. And so to verify that we have our electrodes in the right place before beginning these kinds of long-term recording experiments, we used combined MRI structural imaging of the mouse brain with a CAT scan recording to localize both our electrodes and the fiber optic, so we can effectively phototag the oxytocin neurons in virgin females. You can see one nice phototagged animal here. Then we put her in the cage with a mom and the babies, turn the cameras on and the recording system on and record for days, if not longer. And then the goal is basically to use the spiking activity of the oxytocin neurons to look to see what's going on in the video. These are thousands of hours of video recordings, some of which have been analyzed by hand, although of course we're using machine learning methods now too as the data sets get bigger. And the idea is we're trying to find social needles in a haystack, maybe transient events that could really be important in shaping social or parental interactions. And we've discovered at least two things, and there's guaranteed to be more in the data sets, and I'll talk about that a little bit towards the end. But the two behaviors that kind of first popped out, the first one we call shepherding, and you'll see why. Basically, the mom escorts the virgin to the nest. So we score a lot of behavior in terms of what's going on in the home cage life. But here I'm only showing you in this ethogram time in the nest in gray, time out of the nest in yellow. And you can see that for a co-housed virgin dam dyad. And the virgin, the mom is kind of in and out of the nest the whole time. But look at the virgin, starting on day 3, she's really exclusively in the nest. And there's a reason for that. The mom basically keeps her in there. Okay. Okay, the virgin is getting a drink of water at a hydropack. See, the mom comes along and drives her on back to the nest. Yeah, it's not a subtle behavior. It happens hundreds of times in the course of co-housing, pretty much for every animal pair that we've looked at. If you co-house your animals, I bet you'll see it too. The shepherding behavior basically helps the virgin kind of overcome her aversion to the nest and the pups. And this shepherding behavior definitely activates virgin PVN and oxytocin neurons. Here are 9 simultaneously recorded units. This isn't a task-based behavior, right? This is just spontaneous behavior. But because it happens so frequently, we can record from the same units over and over again and basically build PSTHs. So this first unit, unit 1, you can see it's activated as the virgin is being shepherded. Time 0 is the virgin head nest entry. Units 2 and 3, and unit 3 is an optically identified oxytocin neuron. They burst when the virgin's head enters the nest. And it's a period of time when there's a lot of chattering going on with the pups in the nest. And so there might be a natural pairing then of oxytocin release and the sounds of the pup calls to sort of build representations of pup calls, help the virgin kind of understand what these cues mean. Okay, so the other thing we noticed is that every now and then also the mother herself would spontaneously retrieve a pup. Here now the mother is getting a drink of water. You can see a pup fell off of her. The virgin is now in the nest, and she's sitting there watching the mother perform this spontaneous pup retrieval. And that demonstration of retrieval by the mother activates virgin PVN and oxytocin neurons also. About a half to a third of virgin oxytocin neurons specifically increase their firing rates when the mother animal performs this retrieval. It's probably not intentional, it probably is sort of accidental, but it definitely happens over and over again, and the virgin is sensitive to it, and we have other data indicating kind of what features we're sort of looking at and how that might activate the oxytocin system. One interesting thing is that some of these neurons seem to be mirror neuron-like in terms of their qualities. They respond to the act of the mother retrieving, and then if the virgin herself begins retrieving later, the same units, some of them, also are reactivated in that way. About a quarter to a third of them seem to have this kind of reactivation-like quality. And I think it's interesting to note that recently Nirao Shah's lab has reported mirror neuron-like responses also in the hypothalamus, and in this case in the VMHVL, for animals observing and then participating in aggressive interactions, which is quite different and involves the VMHVL instead of the oxytocin system. Okay. So for the rest of the talk now, I want to focus on the biological mothers. All that work has been published. And there's one really sort of interesting aspect of oxytocin release in biological moms, and that is that about half of human mothers, the sounds of crying babies alone can lead to elevations of oxytocin and even a bona fide milk ejection reflex. The usual way in which oxytocin neurons are activated is by infant suckling by a well-described spinal cord reflex arc that leads to input into the PVN. But for about half of human females, there's activation in the hypothalamus as well when they're hearing the sounds of baby cries. And here's some older data from human subjects monitoring plasma oxytocin. You can see that about 5 or 10 minutes before nursing begins, as the infant is crying in advance of nursing, that's when the peak oxytocin elevations occur. So we wondered, could we kind of see a similar phenomenon in mother mice, and if so, sort of map out the auditory circuit by which input gets into the oxytocin neurons, because of course, oxytocin cells in the PVN are part of the classic auditory feed-forward system, right? So a postdoc in the labs of Ivana Valcheva made, in this case, wholesale recordings in vivo from awake head-fixed mothers, because now under head fixation, we can present all kinds of different stimuli under good control, and we can make bona fide wholesale recordings and ask about kind of the conversion of subthreshold responses to spiking responses. And so she also phototagged these cells using a fiber optic and the patch pipette. And you can see on the right one of the oxytocin positive cells and one of the oxytocin negative cells that she recorded from. The purple lines are the periods where she's playing pup call sounds over and over again. And if you look at that oxytocin neuron, you can see after several seconds of pup call stimulation, it switches its firing mode. It's depolarized and begins firing in bursts of spikes, whereas the oxytocin negative neuron didn't seem to respond in this way. For time reasons, I'm going to skip our circuit mapping. It seems that the main input providing the auditory signals to the PVN is a thalamic nucleus called PIL, the posterior interlaminar nucleus. You can see on the bottom right, it also has pup call responses, albeit with short latency. And so the question then for us is, how do you have a presynaptic input, which is responding to the pup call sound on the order of 10 to 100 milliseconds or so, producing an immediate downstream target cell, a response that really lags in terms of when it starts firing, several seconds. And the short of it is that the oxytocin cells are kept under a high level of inhibitory control, but repetitive stimulation seems to lead to a form of inhibitory long-term depression, turning down inhibition and allowing these cells to fire action potentials. And again, for time reasons, I'm gonna skip this, but we can talk about it afterwards if you like. And so in the final bit of the talk then, I want to ask about what all kinds of cues do mothers need to sense to take care of their pups, maybe sort of on beyond cries, or what is eliciting the cries in terms of spontaneous parental care. And so this is the work of a graduate student in the lab, Louisa Schuster, who set out to sort of upgrade our 24/7 monitoring system. She began her PhD studies really kind of right before COVID, and so one of the ideas here was basically to try to pandemic-proof our ability to collect and analyze data. She built boxes around home cages for good environmental controls with some Arduinos and Raspberry Pis to keep humidity constant, to control temperature, to control the light-dark cycle per cage. There's a camera by the side. There's a camera including a thermal imaging camera from the top. And there's a camera under the nest And I'll show you some interesting data from that at the end of the talk. And the hypothesis was, are there adaptations that occur kind of litter to litter? Are there maybe changes in the oxytocin system that maybe help animals be better at parenting and learn from their experiences the first time around, second and third time around? Just what are all the things that first time and single parents have to do? But much to our surprise, actually, there's a whole lot of pup mortality. And some fraction of animals actually didn't seem to perseverate in losing essentially all of their pups. So this is pup survival per litter on the y-axis, and the litter number across 4 different litters on the x-axis. All these animals are continually monitored. So this is about 4 to 6 months of continuous recording. Each dot is one dam's litter. And basically, the group at the top, those are the same animals, litter 1, litter 2, litter 3, litter 4. The group at the bottom, those are all the same animals. They're perseverating on essentially losing all of their pups. And so maybe I should offer a trigger warning. I'm going to show you some aspects of pup mortality. It's not due to aggression. It's not due to mauling. It really mostly seems to be due to hypothermia. And the fact that some of these mothers, we call them low pup survival dams, aren't so good at sort of building an intact nest to sort of keep their pups warm over time. So here is some thermal imaging. You can see the time at the top. This mom did not build a nest at all, and so her pups sort of scattered and began to cool. They essentially cool down to ambient temperatures within several minutes, and then they go comatose and eventually will expire. Here's one pup. Well, I'll move on. So we've tried to really analyze the nest as a structure. But the nest is actually really dynamic. It's 3D, it's very complicated, and there's more to a nest, of course, than just the visual qualities of a nest. A nest is made to protect and keep warm the pups. It doesn't really matter so much what it looks like as long as it's doing its job in terms of thermal regulation. But we use the visual aspect of the nest as a mask to be able to monitor pup temperature, and then more precisely sort of quantify the drop in temperature depending on different sorts of nests and across dams. So here are 3 different dams, and you can see at the top the fraction of pups that survive, basically 24 hours after birth. And you can see the structural integrity of the nest in terms of how well it keeps the pups insulated when the mother makes excursions to leave the nest. The mother has to leave the nest. She will get really hot and has to leave the nest just to regulate her own temperature. And so she leaves every now and then. She also has to go forage and get some water. When she leaves, she disturbs the nest, and the pup's temperature begins to drop. So the mother is constantly sort of needing to kind of re-engage with the pups and re-engage with the nest and keep rebuilding it. And if we look at basically just how often the dams keep engaging with nest rebuilding, you can see here 4 to 5 animals. These are 5 ethograms from 5 different dams. Each tick here simply just is the animal rebuilding the nest at that time. 4 of those animals keep rebuilding the nest basically continuously. One animal sort of gave up and lost all of her pups thereafter. Okay, so I want to show you 3 more things. First of all, is it just that these low pup survival dams, are they kind of destined to be like that? Is there maybe something genetically different about them that leads them to perseverate in this regard? Or can it be remediated or changed in some way? So these are the older data I just showed you a few slides ago. Because 1/3 to 1/2 of animals lose all their pups, it's pretty easy to get a population and ask, what can we do with them? We identified animals lost all of their pups in a separate cohort, and then split them into 2 groups. One group we co-housed after their second litter with an experienced high pup survival mom who didn't lose all of her pups. The other animals we kept isolated. And you can see after co-housing, for the 3rd and 4th litters, when she's back being a single mother by herself, now that low pup survival dam, all of her babies, or most of her babies, live, whereas the other ones keep perseverating and losing all their pups. This isn't just an effect of social buffering. If we co-house with a virgin female or a male, there's no effect. So what's going on during the period of co-housing? Well, basically a whole bunch of shepherding. Here is, you can tell the low pup survival mom is the one in the bottom corner trying to escape, literally trying to jump out of the cage. The experienced mother keeps pushing her into the nest via shepherding, and here's 24 hours later, an episode of the shepherding, and within 3 days, now the animal up in the nest is that initial low pup survival mom. She'll go on with the next litter she has by herself to keep all her babies alive. Okay. So this documentary approach that we're taking, I think, can really be used for some interesting deep phenotyping and discovery. And one thing that's sort of been confusing in the field for a while now is why can animals deficient in oxytocin receptor signaling even breed? So if you knock out the oxytocin receptor, animals don't have milk ejection reflex, and they lose most, if not all, of their babies. However, most of the times, breeding is done in co-housed animals. And so in our single-parent setup, we sort of examined this, and we studied how oxytocin receptor knockout animals did when they were bred. And essentially, they lost all their pups. They would push in labor for hours and eventually expire. We stopped the experiment for ethical reasons. But if we co-house another female with the oxytocin receptor knockout mom who is giving birth, here's some imaging we've done from under the nest. The animal will clear the cage of bedding, so we get a nice clear view of what's going on. The animal giving birth is a receptor knockout, and the other female comes along and essentially seems to act as a little mouse midwife, helping to kind of pull the pup out and ensuring both the pups and the mom will survive. And so now we're sort of interested in this theory of potential helping behavior and how it is that animals can work together to overcome biological deficits with social adaptations to ensure family survival. And then the last thing I want to say is we really need a repository for video data. We have great repositories for physiology and imaging, Neurodata Without Borders, Dandy, and so on. And YouTube, of course, is not a great repository for scientists. But there is an existing repository really at scale with firewalls, with a legal counsel, and that kind of thing, called Databrary, which is currently for videos of human parents and babies. And we're starting to try to open it up for videos of animal behavior also. So if you're interested in either sharing and getting access to some of the existing videos, or if you have your own video data that you're looking to archive, please come talk to us. This is just the beginning of it, and we'd really like to get going on it. Okay, again, thank you for your patience with all the technical difficulties. We've been doing 24/7 videography to kind of figure out what oxytocin neurons do and other systems in the context of parenting. One thing that's really clear is that even in the sanctity of a lab vivarium, single parenting is a challenge, and now we're working to figure out how we can save and store these data appropriately. I want to thank all the people who've done all this work, Chloe, Haban, Naomi, former students and postdocs, Ioana, Bianca, Jen, Silvana, my other collaborators, and thank you for your attention. Thank you.
So big thanks to Rob. Any questions?
Hi. Really interesting talk. Thanks. I actually had two questions, if that's okay. So first, I was just curious if you had looked at the males at all, because I know there's been like a lot of interesting data about when males do and do not develop parenting instincts, and I was curious if you'd looked at that.
Yeah, so first of all, males have oxytocin receptors, and definitely male black 6 C57 mice can be great parents also. They express interesting paternal behaviors, and in other strains, certainly, Catherine Dulac and her colleagues, among others, have really documented the neural systems that are important for it. One thing that seems pretty clear is that oxytocin supplements don't really seem to help male parenting be expressed any faster. We don't think that's because oxytocin isn't required for aspects of male parental behavior also. We think that actually there are some places, for example, the left auditory cortex, that have more receptors in females, so there's just a higher ceiling potentially for receptor expression and activation to kind of push the behavior along faster. The males can definitely be great dads too.
And a follow-up question, this is like very minor, but I was just curious about what the nesting behavior looks like in, okay, it's like weird to talk about evolutionary context for C57s that have like evolved in labs for however many generations. But I know that in the wild, mice tend to burrow, and I was curious if that affects the structural integrity of the nest. Like, is this maybe different than what you would see in the challenges of mouse parenting in the wild?
Oh yeah, that's a great question. So what goes on outdoors, and what does nesting and burrowing look like outdoors for black 6 mice or other kinds of mice too? First of all, Hopi Hoekstra has done lots of super interesting work in deer mice and other animals, asking about different genes and brain areas that might be involved in nest building and the like. But honestly, we just don't really know what parenting and co-parenting is like in wild mice populations. And I think one thing that's clear from the videos we've collected is we still don't really understand what parenting is like, even in black 6 mice hanging out in Vivaria. There are opportunities. There's a facility called Stony Ford at Princeton where Andrea Graham is rewilding lab mice, taking black 6 mice, putting them outdoors for 2 weeks for the summertime, and then asking what kind of behaviors do they engage in. And so we began collaborating with her, basically dosing it with trail cams. We've got a thermal imaging drone, and the goal is basically to kind of relate nesting and co-parenting behaviors outdoors to what goes on in the lab. There are definitely going to be some differences.
Thank you.
Hi, Rob. Really amazing work. The mouse doula thing is just incredible. My question is regarding what are the qualifications of a mouse doula? Did they require experience? What happens? Can an oxytocin receptor knockout mouse be a mouse doula?
Yeah. Great question. So we want to know all those questions. We want to know the answers to all those questions also. This is sort of brand new stuff. And even so, the identity, the relationship between the two animals, I think it's a reasonable hypothesis that there might be some oxytocin signaling required in the other co-housed animal, who was a wild type so far in the data we've been collecting. And so if it's 2 receptor knockouts, would there be a difference in their behavior? Are there signals that the pregnant dam is emitting that the other animal uses to come and help her? Is she actually helping all that much? And honestly, I think we still have a lot to learn about the basics of parturition in mice and sort of what's required for successful parturition. It's sort of amazing that there's no redundancy in the system. If you knock out oxytocin receptors, other systems don't take over. The milk ejection reflex really does go away. Uterine contractions really are just totally flat. They're gone. And so what are other kinds of adaptations that can occur to help the animals along?
And sorry, I'm going to do a follow-up question as well. So does that imply that the result from Neural Shah's lab, which is what I thought you were going to talk about when you said Neural Shah, with the oxytocin receptor knockout, making everything be fine? Animals can just pair bond, they can mate, they can... So you think that the difference is because they're co-housed?
You're talking about the Brenzen paper from the Manoli lab with Neural also in the knockout voles?
Yes.
Yeah.
Sorry.
Yeah, great. Thanks for bringing that up. So maybe some of you have heard about this paper that came out recently from the Manoli lab at UCSF, making voles that are deficient in oxytocin receptor signaling. They just knocked out the receptor. And so that paper is full of really interesting data. I really love this paper for a few different reasons. First of all, what the paper is looking at is an assay of partner preference. And they don't find much difference when you knock out the receptor compared to wild types, which seemed like it was kind of a surprise because of the belief that oxytocin signaling was important for affiliation and pair bond formation in the vole. The one thing that I think is worth noting is that a partner preference assay is not partner preference, it's not social interaction or attachment more broadly. It really is just a lab assay, right? I mean, especially for y'all here at Cosyne, I don't have to tell you, right? A model of a thing is not the thing. And so lab assays are really useful for phenotyping, but at some point you kind of need to go beyond the single assay and move into, for example, measurements of spontaneous behavior over extended periods of time to figure out what these animals are actually doing. One important thing about the knockout voles, though, is that about half the litters die. And even some of the mothers, there's a high level of maternal mortality in the knockout animals, as reported in the method section. And I think it's important to realize that if 50% of the animals are being lost, if the pup loss is half of in the wild types, that's a shocking statistic. That's not just some behavior going down by about half. That's literally like 50% of the babies are dying. Now the reason there's some pup survival in the vole and there's 0 pup survival in the mouse is that oxytocin is important for milk ejection. It's not important for milk production. That's a different set of hormones. But the baby mouse mouth is just not strong enough. And so the mother with suckling the milk is ejected into the pup's throat, whereas the baby voles can be strong enough to physically pull the milk out, and so half of them live. Sorry, I know that was a really long answer, but I think
I loved your answer. Thanks, Rob.
Discuss. Thank you.
We have time for one more question.
Oh, yeah. Hi. I was wondering if you tried the oxytocin stimulation in mothers, and if there's a difference when the mothers are attending their own pups versus attending pups that belong to a different mother.
Yeah, so one thing we actually haven't done is just given dams oxytocin and asked what happens. It might be that with the low pup survival dams, in terms of the single mothering behavior I showed you at the end, maybe that would help kind of convert them into high pup survival dams. We're definitely thinking about that.
And for stranger pups?
Oh, yeah. So what I showed you in terms of the alloparenting behavior, those virgin females are unrelated to the mother. Those are definitely pups that aren't hers, that aren't related to her. And so back to this issue of how do animals actually parent and co-parent in colonies, it's highly likely that if you happen to be a mouse and there's some babies around, they probably are related to you. And so mother animals, father animals, they will fetch pups that are basically somebody else's. It really is about the vocalization and not the identity of the individual pup.
Okay, well, thank you so much, Rob. And next we have Andrew Bailey from MIT.
All right. Thanks so much to the organizers for this opportunity. My name is Andrew Bailey. I'm a PhD student in Michael Fee's lab at MIT, and today I'm going to talk to you about a neural clock underlying the temporal dynamics of an auditory memory. And I want to start by highlighting something I think is really striking about auditory memories and musical memories in particular. This is a post from the website Reddit, where a user had a song stuck in their head and they couldn't remember the name. Now, despite not being able to remember the name of the song, they could remember the musical content of that song in such great detail that they recorded a 45-second reproduction of that song with 5 different instruments, right? And so this is a really remarkable example, but this type of auditory memory, these earworms, are really prevalent in humans. And in contrast to our perennial discussions about the fallibility of human memory, in this case, it can really feel like you have a tape recording in your head of that song that's being played back in real time. And while I'm not going to tell you how this works in humans, I'm going to tell you about how we were inspired by this analogy to the tape recorder to develop and test models for how songbirds' auditory memories get their temporal dynamics. Now, in the Fee Lab, we study the courtship song of the male zebrafinch. You can see a cartoon of one such bird here, and to the right, you see a spectrogram of his courtship song. If you're not familiar with these plots, time is on the x-axis, frequency is on the y-axis, and the color shows the intensity in a given frequency band over time. You can immediately see that these songs are composed of discrete elements we call syllables, and these syllables form repeating motifs. We'll see if you can hear what it sounds like. All right, so these songs are actually learned at an early stage of development when a juvenile male pupil listens to the song of the adult male tutor and forms a memory of that song. Now, sometime later, that bird will begin to produce its own vocalizations, but at first, they sound very disorganized. However, after several weeks of practice, the bird can produce a song that's quite a good match to the tutor song. And you can compare that here to the tutor song again. All right, so in this talk, we're going to focus on what happens during this very first stage as the juvenile male forms a memory of the tutor song. But first, I'm just going to highlight some key results in the study of the adult song production and song learning. And in particular, we're going to focus on a cortical premotor region called HVC. Now, HVC is essential for production of the adult song and is an integral part of the song motor pathway. It projects to song motor cortex, which in turn projects to the brainstem to produce the song. Now, early work in the Fee Lab recorded the neurons in HVC that project to the motor cortex to try to understand their role in the song. And this is what they found. So here you see a spectrogram again of the bird's adult song, and below you see recordings of different neurons in HPC with each neuron shown in a different color and each row showing a different rendition. And what you can see is that each neuron fired a tight burst of spikes at one time in the song, and together they formed a sequence that tiled the song. Now this led to the formulation of the following model, in which the dynamics within HPC generated the timing of the song, and the pattern of synapses downstream generated the content of the song. Okay, so this is a nice idea, but how can we test it? Well, previous lab members reasoned that if the dynamics in HPC controlled the timing of the song, then manipulating the timescale of those dynamics ought to change the timescale of that song. And to do this, they developed a Peltier-based thermoelectric cooling device that could be attached to HPC to manipulate the temperature of this brain region during singing. And here I'll show you what they found. So this is an example of a bird implanted with one of these devices with HPC held at its normal temperature. But what they found was that as they cooled, the song stretched out more and more in an approximately uniform manner. And when they heated, it even sped up slightly. And altogether, this led to an approximately 3% per degree relationship between song speed and temperature. Okay, so we think that this is kind of our best evidence for this model where the dynamics for timing are in HPC. And already you can see this analogy to a tape recorder, where HPC is like the motor of the tape recorder, and the content stored on the magnetic tape is like the pattern of synapses downstream. Okay, so this is HPC's role, we think, in the song production. But how is the song actually learned? So a huge number of studies across many labs have delineated this set of regions shown in green that's thought to perform reinforcement learning during song learning. And this reinforcement learning is thought to depend upon error signal that's been observed in VTA. Now, subsequently, this error signal was tracked back through the auditory system to a region that's interconnected with HPC, leading to the question, does HPC also have a role to play in the formation and recall of the tutor memory? And indeed, a set of recent studies, including two very nice studies from the Mooney and Roberts lab, showed that auditory inputs to HPC during tutoring are required for accurate imitation, and the auditory output from HPC back to the auditory system is also required during subsequent learning. And so it seems that in addition to HPC's function as a motor clock during song production, it also has a role to play in the formation and recall of the tutor memory. And here we're going to ask, what is HPC's role in this process? And what, if anything, does this have to do with its function during song production? And we started with a very simple hypothesis, which is that maybe HPC acts as a clock for the tutor memory, just as it does during the production of the adult song. Now, what are the consequences of such a model? Let's zoom in on this tape recorder analogy. So here you can see two reels of magnetic tape that are turned by a motor across a read head and a write head. Now during recording, sound comes in and it's inscribed upon the tape, on the magnetic tape by the write head. And during playback, the motor turns the tape at the same speed past the read head to produce that same song. Now, of course, like I showed you before, if you slow down the motor during the playback, the song should slow down. But what happens if we slow the motor just during the recording phase? So sound will still come in, it's inscribed onto the tape, but because the tape's moving more slowly, there's less tape between each note. And thus, when you play it back, the song will sound sped up. And so we hypothesized that if dynamics in HPC control the timing of the recall of the tutor memory, that cooling during tutoring will lead to a sped up memory, and thus to a sped up imitation of the tutor song. And to test this, we designed a new variation on this thermoelectric cooling device with a modular design allowing it to be removed up to the end of tutoring and using PID feedback on a thermocouple. And we implanted birds with this, and I'll just show you a few examples. So I'm going to show you an example of a bird cooled by 5 degrees C. This is just the tutor song. On the left, you see a spectrogram with 4 different syllables, and to the right, you see a raster of many renditions of this song, where each row is a different rendition, and the intensity shows the sound amplitude over time. So we implanted a pupil with this device. We cooled HPC by 5 degrees C during tutoring. Otherwise, we left HPC at its normal temperature, and we asked, what song does this bird produce when it grows up? And here you see an example from this experiment. So you can see both in the spectrogram and in this raster to the right that the song is compressed relative to the tutor song. And we could quantify this with the syllable duration distribution. So here you see the syllable duration distributions for these blue syllables with the solid line showing the pupil and the dotted line showing the tutor. And you can see that the pupil syllables were sped up relative to the tutor. And you can see this pattern held up for all the other syllables. And we quantified this as the percent pupil song dilation. All right, so we repeated this in a control bird. You can see here, this bird was implanted with the same device, but HVC was held at its normal temperature throughout tutoring. And here you see that there was no systematic change. So we repeated this for many, many birds, for 35 birds, and I'm going to show you the result here. So here's the results for just the control condition, where each point is a different syllable imitation. And the y-axis shows the percent dilation with negative numbers showing faster songs and positive numbers showing slower songs. And the x-axis shows the difference in HVC temperature during tutoring relative to its normal temperature. And what we found is that as we cooled, the song sped up little by little, and when we heated, they even slowed down slightly. And this led to an approximately 2% per degree relationship between the song dilation and temperature. And you can relate this here to the results during song production, where you see the opposite relationship and around a 2.8% per degree relationship. So I want to pause here and just highlight that these birds aren't doing any overt behavior. They're simply sitting and passively listening to the tutor song, yet a change of a few degrees of HVC temperature has this long-lasting impact on their behavior many weeks later. Okay, so it seems that the dynamics in HVC might control the timing and recall of the tutor memory, but what does HVC activity even look like during tutoring, and how does it support this phenomenon? And so we turn next to two different approaches to record activity in HVC. So one was with miniscope head-mounted imaging in freely moving birds, and one was with head-fixed birds using neuropixels. And I'm just going to show you two example birds in the time I have left. All right, so here's a bird that was injected with GCAMP6F and imaged, and you can see two example neurons here. So at the top you see the spectrogram, again you see a raster of activity, and at the bottom you see the average z-squared activity. And what you can see is that these two neurons fired reliably at a single point as the bird listened to the tutor song. And as a population, these neurons tiled the entire tutor song. And we can do the same thing now with neuropixels. And we see, again, examples of neurons that fire sparse bursts at a particular time in the song, and an entire population that tiles the whole tutor song. And so this is quite striking, this resemblance between the adult activity in HVC during motor production and what happens as the bird listens to the tutor song many weeks earlier. And so now in my last few minutes, I'm going to outline a mechanistic hypothesis for how we think HVC can function in the formation and recall of this tutor memory. Now, I showed you that during tutoring, that auditory inputs come into HVC and drive these reliable sequences. Right? This happens over and over, and we hypothesize that this leads to the strengthening of connections between these neurons, forming a sequence. Right? And so once this sequence is formed, we hypothesize that it projects back to the auditory system, where it acts as a temporal basis to cancel or form a negative image of the expected auditory inputs during tutoring. Now, a nice consequence of this is that when the bird then sings its juvenile song, it automatically recalls this tutor memory in a way that's aligned to its developing song. And by comparing this expected tutor song with its actually produced sounds, it produces a moment-by-moment error signal that can be sent to VTA to drive reinforcement learning. And again, you can see here, by analogy to this tape recorder, how HVC is like the motor and the downstream synapses are like the magnetic tape. And with that, I'll just conclude. I showed you how focal cooling can test our hypotheses about HVC's role. I showed you that we observe sparse sequences in HVC consistent with a role as a memory clock. And I outlined a mechanistic framework for a unified view of HVC's role in both memory formation and motor production. And with that, I just want to thank my advisor, Michael Fee, Natasha Denishenko for help with the viral expression, the Fee Lab, as well as Mike Long and Felix Moll for help with head fixation and their funding sources. Thank you.
We have time for 2 questions. Anyone?
Hi, hello? Andrew. Really, really nice talk. So I guess my question is, during the tutor song, you're cooling HVC, but then what about all the rehearsing? You know, the juvenile's going to sing and listen to itself. Is it cooled then too? Or, you know, I guess I'm just trying to understand how long was HVC actually cooled for to make this possible?
Good question. Thank you, Kay. Yes. Was it? Oh, sure. Yeah, so the HVC is only cooled during tutoring. We tutor for 1 hour each day for several days, and it's only cooled at that time. The rest of the time when the bird practices for several weeks, it's kept at its normal temperature. Yeah.
Hi, thank you for the wonderful talk. I had a quick question about the spectral content of the song that's learned. You show this really nice time dilation effect. Have you looked at whether or not that impacts the spectral content as well?
So I haven't looked very closely at that, but it doesn't appear to affect the spectral content. Yeah, thank you very much.
All right, well, let's give Andrew another round of applause. So next we have Nathan Schneider from Pitt. Hopefully.
You didn't want to use this one?
It wasn't working either. I brought my own in case that was the issue. It's not enough on the screen.
Okay.
Let me try.
But you should detect. Yeah, we should detect something there.
This is probably this one?
Yeah, you started that one and it didn't work apparently. Oh, there we go. Don't touch anything. That seems promising. Your screen flashed, which is a good sign usually. This is terrible. Everybody here deserves an apology.
Yeah, I don't know. Doesn't seem to
So
be
does
doing
this mean that it thinks there is a second screen?
I think so, yeah. Can one of the other speakers bring their computer up just as a sanity check?
Andrew, let's use yours because we know it works.
It's been bizarre because we haven't had any problems up until this session. So it is bizarre that it was able to work.
You can actually move this thing around, which is not ideal. So if it's with the box, then we need a new box. Take another laptop. Okay. I'm going to ask Lily. Libby, can you bring your computer?
Yeah, but I'm not successfully connected. I think you're right. Open her up. Yeah,
Well,
this is the
let's
message.
try this.
Ah, there we go. Yeah, she's good.
Okay.
Let's go.
Hey, folks, I just want to apologize again for all the difficulty, but we have Libby's talk working. We're going to work on Andrew's talk. So we'll go ahead and get started again. Thanks, Libby.
Hi, everyone. In recent years, a major focus of computational neuroscience has been on the quantification of high-dimensional behavior. This can be highlighted by Dr. Bob Datta's keynote talk at last year's meeting on MoSeq, or this year's talk, keynote talk by Dr. Mackenzie Mattis on Zebra, and numerous other works at this conference and in the broader field. In this session alone, we've had inspiring examples of how richer and more naturalistic and more complex behavior are important for the study of the brain. And these advances in post-tracking and postural behavioral models are enabling this shift in systems neuroscience. However, these methods are geared towards the relatively short timescales of seconds to hours. We know that neural and behavioral dynamics also develop at longer timescales of weeks, months, and years. But we haven't had the data set or the accompanying methods to do so. With this motivation, I'll be sharing ongoing work about a method analyzing continuous longitudinal behavioral measurements across multiple timescales via a bottom-up approach. I'm fortunate to work with amazing collaborators in the Brunei and Daisaroth labs, particularly Drs. Claire Bedbrook and Ravi Nath, who designed and lead the overarching project that this dataset was collected for. In this experiment, the movements of genetically identical killifish are tracked from adolescence to the end of life using an automated aquarium setup. Fish are individually housed for their 6 to 9 month lifespan, and an overhead camera records a video from the table of tanks. Key points on the fish's body are identified and tracked during the video process. African turquoise killifish are an exciting model organism because they're the shortest living laboratory vertebrates exhibiting all of the hallmarks of human aging. There's also a growing suite of genetic tools that are being developed for them. This situates them as a unique model organism for the large-scale, high-throughput, and close study of aging-related neurological diseases. This is also the first 24-hour, 20 frames per second high-dimensional behavioral data sets of vertebrates taken over their lifespan. And this enables us to ask previously intractable questions about behavioral dynamics over different timescales, individual variability, and long timescale biological processes such as aging. Specifically, we ask, what is the structure of behavior over the course of a day? And how does it change over the course of the months of a fish's lifespan? How variable is behavior as a function of age across a cohort of individuals? And finally, given observed behavior, can we make biological predictions of how long a fish will live? To begin, we first extract behavioral representations at the sub-second time scale. A common way to do so is to use a hidden Markov model, where observed key point features are treated as the emissions of a sequence of discrete hidden states that we call behavioral syllables. Taking this approach is nothing novel per se, since existing methods such as MoSeq and BehaveNet use variants of HMMs under the hood. But I'd like to make a brief note of the engineering feat involved in fitting an HMM to 6 billion frames of data. This required developing a scalable, parallelizable stochastic optimization method, which is built using JAX and the recently released dynamic state space modeling package. I'm happy to talk about this effort offline, but I have several killifish lifespans worth of behavior to get through in the next few minutes. So inference in the HMM gives us a sequence of syllables for each frame. Here, I'm showing a video clip on the bottom where the fish is currently swimming in place. Above are the corresponding syllable labels depicted as a one-hot encoding. And the labels on the side represent rough syllable groupings by posterior similarity of the key point features. Shortly, we'll see the fish reversing, turning, darting, before swimming with interspersed pauses. All these sub-second and second-long movements are captured by the syllables. Now, this was just a 30-second clip of a much longer recording. If we zoom out to 10 minutes, we find that the fish spends the predominant amount of its time in this window like it did in the latter half of the clip, where it was turning, darting, and swimming. We can summarize this window of behavioral syllables by a count of syllable usage as shown on the right. Now, zooming out to 10 minutes, we find that we can see the circadian rhythm and the fish's behavior begin to emerge, with a significant increase in higher activity syllables that are situated on the lower part of the y-axis between the daylight hours of 8 a.m. and 8 p.m. And now this day of recording is situated within the 260 days of this fish's lifespan. The striated pattern that you can see is the circadian rhythm that we had previously been zoomed in on. It's relatively easy to see some of the large-scale changes in overall syllable usage across the 3 regions of life that I'm pointing out. However, it's difficult to have a sense of the more subtle changes across life. Now, when we look across fish in a cohort, we also see distinct behavioral variability across individuals despite their identical genetic makeup and environment. And now it's even more difficult to grasp what representation, from these representations, what are the common drivers of biological aging? So the central methodological questions here are, how do we model the within-day dynamics of syllable usage over life and across individuals? Specifically, how do we go from syllables, which are representations at the scale of seconds, to representations at the minute scales, to days, months, to understand the book of life? Let's first consider how to go from the scale of seconds to minutes. The naming of stereotype behavioral motifs as syllables originally drew its inspiration from natural language. If we take this analogy further, we can look towards language modeling approaches such as topic models. Topic models, also known as latent Dirichlet allocation, is a highly successful probabilistic approach to natural language modeling. It describes a document of words as a mixture of topics, shown on the histogram on the right. Each topic is a distribution over the vocabulary of words in the corpus of the documents depicted on the left. And importantly, a single document can be about multiple topics with different proportions. Now, in the behavioral setting, we treat a window of syllable labels as a document. Each document is a mixture of behavioral topics. These topics are distributions over the syllables, which are again roughly sorted by posterior feature similarity on the x-axis now. So for example, roaming in pink is described by straight swimming and swimming at the edge of the tank with short pauses. Feeding in green is described by bouts of pitching, flips, and quick darts. And deep sleep is described by inactive and pausing syllables. Now, during this 10-minute window, if we look back to the right, 70% of the syllable activity arises from roaming and darting, and the rest is largely accounted for by drifting, swims, and feeding. And note that the deep sleep topic has 0% probability here, which is consistent with the bin being taken at 12 p.m. right before food is being dispensed. These behavioral topics and their associated usages provide a lower dimensional representation of a time bin of syllables. Now, a time bin has another time bin next to it. And as we begin to zoom out from a single time bin to an hour to a whole day, we can again begin to see the within-day temporal structure that we had picked out by eye earlier. Notably, we see on the left that during the nighttime, usage is dominated by different sleeping and pausing topics. During the daytime, there is strong upweighting of the roaming and darting topics, and relative changes of usage are also driven by when the food is dispensed. Now we ask, how does behavior change across lifespan? When we look across life, the topic usage dynamics we've just shown were at 105 days, and if we look earlier in life, at 50 days, the topic usage dynamics exhibit similar circadian stimulus-driven structure, although with greater probabilities of roaming. However, during the geriatric state of life, the topic usage dynamics are markedly different, with significant dysregulation in circadian rhythm. To quantify these changes, we can embed the topic usage dynamics. We can take out this temporal structure and embed the reduced topic usage dynamics in even lower dimensional space of aging-related factors. So here on the right, I'm showing 2 dimensions of the space for visualization purposes. The triangle marker denotes the start of recording. We see the trajectory advance through space with the 50 and 105-day dynamics embedded in their indicated locations. And in early life, there is that relatively rapid traversal through the space before stabilizing briefly during midlife. The age factors eventually traverse out of this space and enter a geriatric stage of life, where again the behavior is relatively stable before entering a pre-death stage. Now, this is the aging trajectory of a single fish. When I layer on more fish, we see that there's considerable variability in the actual topic usage, but at the same time, they follow similar counterclockwise trajectories through the space, with brief periods of stability in midlife and late life. Now, I'll continue to layer in additional fish who have different length lifespans. The yellow are longer-lived fish, and the blue that I'll be showing are shorter-lived fish. In all of these cases, we find that fish in the cohort follow the same aging trajectory. The shorter-lived fish appear to die suddenly along this trajectory, while the longer-lived fish have a more gradual progression into the geriatric state, but seem to live extremely long because they are spending a considerable number of days in the near-death stage. This 2D visualization demonstrates the potential for these aging factors to offer a lens for predicting future behavior and lifespan. Our preliminary hypothesis is that the fish in a given cohort and environment follow the same underlying canonical trajectory through aging factor space. We parameterize this with a Gaussian process. We then test this hypothesis by holding one fish out and training the proposed model on the rest of the cohort. Here, I'm showing two aging factors in the red and purple across age of the held out fish, which we treat as observations. Using the fitted GP, we compute the posterior distribution on the initial set of days shown in the lighter blue region. Then we can use the conditioned Gaussian process to predict the future trajectory shown in light gray to the right of the blue region. Using this allows us to get at our final question of, can we predict lifespan? Here, we sweep over the future possible values of when the fish would die and compute the marginal probability of this date under the conditional Gaussian process. The white vertical line denotes the true lifespan of this fish at 249 days. We see that the predicted probability over lifespans in blue has a mode around 250 and 280 days. Now we compare this model to the empirical distribution of lifespans in the cohort and treat this as our baseline model. As the Gaussian process is conditioned on more observations, we see that the precision of the GP prediction distribution increases while the baseline model can only make a naive guess. This points to the predictive power of age factors derived from topic usage dynamics and opens an exciting possibility for online lifespan prediction. So that's been a whirlwind of a talk. To summarize, we have extended a topic model to vary dynamically over time bins within a day. We find this topic usage to be strongly structured by environmental stimuli, but it breaks down consistently across individuals with age. We can extract aging-related factors from these topic usage dynamics across individuals that are interpretable and predictive of lifespan. This has all been possible due to the incredible continuous whole lifespan killifish behavioral dataset. Our questions so far have been focused on the behavioral side. Looking forward, we're interested in understanding changes in neural and behavioral activity in fish that have been genetically modified to model humanized neurological diseases. And while our model has been motivated by this whole lifespan dataset, we see much more general applications of it to neural and behavioral datasets that are collected over multiple sessions, and I'll be excited to talk to you about all the possibilities. Thank you. I would like to thank my amazing team of collaborators on this project. Amber Hu, who's in this audience, is an up-and-rising graduate student in statistics, and she's been an absolute powerhouse on the project, and particularly with dynamical modeling. Doctors Claire Bedbrook and Ravi Nath, who, as I mentioned before, are the real drivers of this project, and they truly epitomize the breaking new ground in straddling multiple disciplines, in particular Claire, who's been a key contributor to the model development since day one. Both Claire and Ravi will be on the academic job market in the upcoming year, so please keep an eye out for them. And finally, thank you to Carl, Ann, and Scott, who have been invaluable scientific advisors throughout the entire process of this project. I'll take questions now.
Thanks so much, Libby. That was fantastic. We have time for one question.
How's it going? Very nice talk. Have you been able to quantify the probability of transitions between different topics and syllables and how that changes over a lifespan?
Right. So we model the dynamics from within days with a weighted combination of basis functions. And then we are able to kind of pull out those basis functions and get a reduced topic usage. And then the Gaussian process modeling that I was showing you is modeling the dynamics across the days. So we have kind of two different sets of temporal structures.
Sorry, I should have kept Libby up here for another second. But that's okay. Thank you, Libby. Hopefully we'll get this straightened out. Again, I apologize.
Try your computer. Thanks from you. Okay. Yeah.
We're still not getting a connection there. It seems to be that it's working on that. She was in that as well. Good. So yeah, so we're gonna get.
Yeah, let's go.
Let's get
We might have.
Yeah, come on.
It's now Rob's computer doesn't work either, but it might be a Mac versus PC thing.
Okay.
So if that's the case, we're just gonna go. Yeah.
Okay.
No. We're gonna just. If this works, it's not even.
It didn't register it. Normally it will flash.
Hey, folks, so we're going to move ahead with our last speaker, Kate Hartley. And I just apology again to Nathan Schneider. We're going to find another time for him. So, Kate, thanks for coming.
Okay. Great. So thank you to Jess and Blake for the invitation to be part of this program. It's a pleasure to be here and to share some of my work. So I'd like to start with this video, which I think reflects very nicely the fact that from the earliest stages of life, humans are avid explorers. We actively engage with stimuli in our environments, and even though the actions that we may deploy may not be directed toward achieving specific goals, they still serve an important adaptive function. Exploration is critical for discovering the affordances for action in our environment and for learning about the structure of our world. So, ooh, sorry. All right, now I'm... So, we continue to explore throughout the lifespan, even as we go about goal-directed behaviors, increasingly so as adults. Because of the inherent complexity that's present in our environment, we're constantly encountering situations that are novel and uncertain. And while it's clear that many aspects of exploratory behavior change with age, it's not well understood what drives our exploration, what effects exploration has, and how these factors might change systematically across development. So that topic is what I'm going to tell you about today. And while the vast majority of my research uses controlled tasks and laboratory measures to investigate the neural and cognitive and computational mechanisms that underpin learning and decision-making across development, today I'm going to tell you about some more recent work that is merging these laboratory-based studies with measures of real-world behavior. And this is really our lab's first foray into studying motivated behavior in the real world. So today, in this body of work, I'm going to attempt to show you through merging real-world measurements and laboratory studies that exploration is subjectively rewarding, that exploratory behavior, the tendency to explore, exhibits systematic changes over the course of development from childhood to adulthood, and that the cognitive determinants of exploratory behavior shift with age. Okay,
So exploration being subjectively rewarding. So I'll give you an anecdote. I live in Brooklyn, New York. When I leave my home, I typically take the subway to NYU where I work. If I take a different route home and maybe after walk through the park, hop on a city bike, pass some taxis, ride over the bridge, I'll see different sights and sounds in the course of that novel trajectory. And if I hop on a plane and fly to Montreal, I may see entirely novel sights that I've never seen before in my life. And the thesis of this section of the talk is that the novelty and experiential diversity that we encounter as we navigate our environments is subjectively rewarding and promotes positive affect. So the notion that experiential diversity promotes well-being has been demonstrated quite convincingly in studies that explore the effects of enriched environments on animals' development. So animals that are raised in environments in which they're able to roam freely, in which they have access to novel objects, to conspecifics, exhibit a range of behavioral outcomes that are consistent with the notion that it's promoting that's having reward-like effects. These animals play more, they show higher levels of social affiliative behavior, they evaluate novel situations optimistically, and they show resilience to stress. Rearing in enriched environments also leads to a host of neurobiological plastic changes in regions, sensory and motor regions, including cognitive regions associated with learning, including the striatum and the hippocampus. And it's been proposed that the effects of enriched environments may stem from the subjectively rewarding, at least in part, from the subjectively rewarding properties of novelty. So novel stimuli can reinforce behavioral responses, elicit activity and reward-sensitive dopaminergic circuits, and promote preferences for the contexts in which they're encountered. And we know a great deal about the neural circuits that are involved in many of these effects. Through engagement of a hippocampal VTA loop, exposure to environmental novelty can drive release of dopamine, and this has beneficial effects for learning and memory, and may also underpin the subjectively rewarding properties of environmental novelty. So in the first study that I'll tell you about today, we wanted to ask the question of whether exposure to real-world experiential diversity promotes positive affect in humans. And this work was really inspired by a study that was published in 2013. Julia Freund is the first author. This was work done with in Gerd Kemperman's lab with Ullman Lindenberger, Andreas Brandemeyer, and others. And in this study, mice were reared in genetically identical mice, which made this a particularly insightful piece of work, but were reared in a large enriched environment. Each animal was implanted with an RFID tag. And throughout the environment, there were RFID sensors that enabled triangulation of where the animal was in the environment. Within the enriched environment, there were different levels, there were conspecifics, there were wheels, novel objects, bedding, which meant that if animals explored their environment, they had the opportunity to come into contact with environmental novelty. So importantly, animals showed different behavioral patterns within the environment. Animals who demonstrated lower measures of what was termed roaming entropy. So this is just the distribution of animals' time across space, distribution across space over time within the environment, either way. So an animal with low roaming entropy, you can see here, shows spending a lot of time in one location in the environment, maybe makes an excursion to another location, but most of the density of their time is spent in one region, whereas an animal with high roaming entropy is showing greater coverage of their environment. And what was found in this paper was that higher roaming entropy animals showed sort of recapitulation of many of the neural and behavioral consequences of being reared in an enriched environment, including elevated levels of hippocampal neurogenesis. So here we wanted to ask, does exposure to experiential diversity in humans promote have beneficial consequences and promote positive affect. We all carry around in our pockets devices that are capable of continuous geolocation. We had participants in New York City as well as in Miami. This was done in collaboration with Dr. Aaron Heller at the University of Miami. We recruited participants to test this hypothesis. We had participants come into the lab. We configured the apps on their phone. And for 3 months, we did continuous geolocation recording. And every other day, we asked participants to rate on a 10-item quick slider scale their degree of positive and negative affect. So we found that roaming entropy of our participants varied from day to day. Here's one day, a low roaming entropy day of a participant in Manhattan where they have one modal location and maybe make one excursion over the course of the day, and another day where they're covering a lot more territory and spending less time in that same modal location. Here we have a participant in Miami on a high and low roaming entropy day. And we can quantify the distribution of the minutes of a day across locations in space to derive a measure of roaming entropy. And we asked, do daily fluctuations in roaming entropy relate to fluctuations in affective state? And we found that yes. On days where roaming entropy was higher, participants reported higher levels of positive affect. Roaming entropy was unrelated to negative affect. And importantly, this effect holds when we control for many factors that might be related to either affect or movement, including distance traveled, city, time of day of the week, temperature, precipitation. So we wanted to know whether there might be evidence that the hypothesized circuit that I told you about in the brain might play a role in this relation between entropy and affect. We brought back as many of the participants as we could, and we had them undergo a resting state MRI scan. From these data, we defined a seed region within the nucleus accumbens of the ventral striatum. This is a region that in many studies across species has been associated with a response to reward and environmental novelty, as well as with positive affect. And we asked, where in the brain does the strength of the correlation with the activity in this ventral striatal seed region relate to the strength of an individual's relationship between roaming entropy and positive affect? And we found that individuals who exhibited a tighter correlation between ventral striatal and hippocampal activity showed a strong, for those individuals with a stronger correlation, roaming entropy was associated with larger increases in their positive affect. Okay, so I've been talking to you about constructs like novelty and experiential diversity. But I haven't really told you about whether the roaming behavior is bringing individuals into contact with novel locations. An individual could have equivalent roaming entropy on a Monday and on a Tuesday and visit the same number of locations. Or have higher roaming entropy and visit the same locations they've been in the day before. So we asked, is roaming entropy really associated with heightened novelty exposure? We took our data, and for every participant, we held out their first 10 days of data and identified every location that they had visited in those first 10 days as old. Then, starting on day 11, any location that had not been present in the first 10 days was coded as a novel location. We quantified the number of novel locations visited on every day, and we found that indeed, visiting a greater number of novel locations was associated with heightened positive affect, and that the effect of environmental novelty was even more robust than the effect of roaming entropy, which is novelty agnostic. I've also been telling you about experiential diversity, but you might ask, on days of higher roaming entropy, are people actually having more diverse experiences? There are large swaths of New York City or of Miami that may be perceptually similar or have similar sociodemographic features of the environment. Are these days of heightened roaming entropy really more experientially diverse? To answer this question, we took every GPS coordinate that was present in our participants' data in both New York City and Miami. And we pulled US Census data capturing about over 100 measures of sociodemographic features of the environment at a level of resolution that is called a block group. It's the unit of space that 500 households live in. So it's smaller than a zip code, but larger than a block. Well, it depends in Manhattan. And we did a principal components, and this includes data like the educational attainment, the ratio of businesses to residential homes, the mean age of the people living there, median incomes, gender. We did a principal components analysis on this data and found that the first 3 components accounted for about 85% of the variance in the measure. Then, for each of the top 3 principal components, we discretized these components into high or low levels, okay? So for every location in space, we were able to recode it as being in a position in the sociodemographic feature space, high or low on PC 1, 2, and 3. So now all of New York City and all of Miami is depicted here as one of 8 locations in sociodemographic feature space. And we asked the question, is entropy over this 8 location space of variation in sociodemographic features associated with fluctuations in positive affect? And the answer was yes, even more strongly so than for roaming entropy over GPS coordinates. Okay. And I should note that the 2, roaming entropy and entropy over the sociodemographic feature space, are not trivially related. You can see that there are huge swaths of lower Manhattan, for example, that are coded as a single location. And so a day that an individual spent in that region of space would be considered a single location in this analysis. All right, another question you might have is, what's the causal direction of this relation? I've been telling a story which was the direction in which we hypothesized these results might be strongest, that exposure to novelty would drive positive affective states. But it could well be the case that on days when we feel happier, that we're driven to explore our environments more. And while we were not intrepid enough to attempt to causally manipulate roaming behavior or affect in our participants, we did take advantage of the long longitudinal time series data that we had and conducted lagged analyses. So we asked, does yesterday's novelty and diversity predict today's positive affect, controlling for the same day relation? Does yesterday's positive affect predict today's exposure to novelty and experiential diversity? And the answer in both cases was yes. So this suggests that indeed there are bidirectional relations. And I came across this figure while we were in the course of writing the paper. It's from a social psychologist named Barbara Fredrickson, who proposed that there's a sort of upward spiral that positive emotion engenders, where positive affect drives us to explore greater novelty. This has a number of benefits that increase positive affect and perpetuate this cycle. And here, this bidirectional relationship provides some support for this concept. Okay, so far I've told you that exposure to experiential, novel and diverse experiences is associated with fluctuations in positive affect in humans, that the strength of this effect is associated with a degree of functional correlation between the hippocampus and the ventral striatum, regions involved in novelty and reward processing. And that the relationship between entropy and affect appears to be bidirectional. Okay. Next, I want to go on to explore whether exploration exhibits systematic age-related changes. There are strong reasons to believe that this might be the case. So there are many proposals that the sort of undirected play that children exhibit at earlier stages of life reflects a sort of systematic change between exploration and exploitation across the lifespan, and that a bias toward greater exploration at younger ages might promote better learning about the structure of our environments. And there's evidence not only from humans, but from many other species to suggest that that's the case. This is exploration of novel objects, either simple or complex objects, in juvenile and adult raptors. And here we see that a greater proportion of juveniles are engaging with these novel objects. Similarly, in 2 species of non-human primates, you see greater engagement with novel objects. There's also data suggesting that exploratory behavior might not just linearly exhibit linear age-related changes, but there might be nonlinearities, particularly during adolescence. So here are adolescents being more willing to enter the open arms of an elevated plus maze, which rodents often find anxiogenic, or they're reluctant to do. So adolescence is a developmental stage that is associated both anecdotally and in the scientific literature with heightened reward sensitivity and increased exploration. And it's been proposed that novelty seeking may be a driver of exploratory behavior based on laboratory measures like what I just showed you. Further, there are functional accounts that propose that heightened exploration during adolescence may facilitate the transition toward independence by helping to promote peer affiliation and helping adolescents sort of scaffold their ability to make autonomous decisions in new environments. So here we wanted to use the same approach to ask the question, does exploratory behavior operationalized as roaming entropy change systematically with age from adolescence into adulthood? We had 28 adolescents here, 13 to 17-year-olds, 30 adults, age, I believe, 18 to 27. I'll show you in the next slide. Come into the lab, and we use the same geolocation tracking approach, asking them every other day to rate their affect. We also collected measures of self-reported real-world risk-taking and social interaction data to test some of these hypotheses about the potential consequences of exploratory behavior. We found that roaming entropy did indeed exhibit systematic changes with age, here increasing across the teenage years, potentially showing a peak around the age of 18 to 20, legal independence. And I wouldn't make much of this decrease. Our sampling density in that young adult range isn't sufficient to really draw that inference, although there are mobility data that extend into adulthood and older adulthood that suggests that indeed there are peaks in in adulthood and in mobility that decrease with further aging. So we found that as in adults previously, roaming entropy was associated with greater positive affect. But while the individuals showed these positive slopes, there were no differences in the strength of this effect as a function of age. So this effect was equivalently strong in adolescents as in adults. When we look at our social network connectivity data, we found that when we look at the relationship between individuals' mean roaming entropy, so this is averaged across all the days in our sample, individuals who explored more across both adolescents and adults had larger social network sizes. We next turned our attention to the self-reported risk-taking data. And we found that within our adolescents, selectively, there was an association between heightened roaming entropy, so individuals, adolescents who explored their environment more on average, reported higher levels of risk-taking, but this effect was not present in our adult participants. And I'll speculate about that in a moment. So in this study, we found that mean levels of roaming entropy, our proxy for exploratory behavior, exhibited systematic changes with age. We also saw a hint, so we really expected this peak in exploratory behavior might be earlier in adolescence, consistent with the idea that maybe biological drivers of exploration that are associated with puberty might be driving these effects. Instead, we found that the peak in exploration was shifted to about 18 to 20. We're interested in the idea that sociocultural factors may modulate the expression of exploratory behavior. This is a paper looking at exploratory behavior from childhood to adolescence in Hadza youth. This is a modern hunter-gatherer people that live in northern Tanzania. And you can't really read the axis here, but it's the length of the distance traveled, the furthest distance away that they go, and you see systematic increases in these measures from age 8 to 13, 14. And this is just to illustrate that cultural norms governing the roaming behavior of individuals across age may also be powerful determinants of this sort of exploratory behavior. So we're interested in probing this interaction. We also found that we replicated the effect that day-to-day fluctuations in roaming entropy were associated with positive affect, but we found that the strength of this effect was age invariant. We had hypothesized that potentially it would be the case that increased sensitivity to novelty might make exploration more rewarding for adolescents, but instead we found age invariance in this effect. When we conducted our individual difference analyses, we found that individuals who explored their environment more, whether adolescents or adults, had larger social networks. And again, this begs the question of the causal direction of this approach. It could be that this reflects a common drive for diversity in experiences, including in social interaction partners. It could also be the case that individuals with more friends have more opportunities to have different places to be. We're interested in the way that the evolution of this unfolds over time in future research. However, we found that the association between exploration and risk-taking was specific to adolescence. And one of the ideas or sort of speculative interpretations we have here is that exploration during adolescence is different than exploration during adulthood in that for many adolescents, this is the first, this is the beginning of the stage of their life in which they are encountering novel environments without parental supervision. And they may be encountering novel classes of environments, going to a party somewhere with peers where they're not parentally supervised, going to places where there are opportunities to drink alcohol or experiment with drugs. When adults encounter these situations, they do so with well-refined decision policies about how they behave in these contexts. And it may be the case that adolescents who are encountering these contexts more because they explore more, may be behaving in ways that are risky, but this association breaks down into adulthood as individuals refine their decision policies for how they behave in these contexts. Okay, so these data suggest that exploratory behavior changes systematically with age, but these data present a confound, which is what the next laboratory study attempted to disentangle. So I've been talking about encountering novel environments and treating the effects that we're observing of being in novel environments as effects of novelty. But novelty and uncertainty are inherently confounded in many situations. When we encounter perceptually novel objects, when we encounter situations that we're unfamiliar with, they both may be perceptually novel in their appearance, but also uncertain in the affordances for action or the reward value that they present. And that was what we attempted to dissociate directly in the next study I'll tell you about. So now we're shifting into the laboratory where we recruited children, adolescents, and adults age 8 to 25 to do this study. We had participants do a task in which they're told, you are searching in hiding places in a magical kingdom where gold coins have been hidden. Your task is to find as many gold coins as possible. They're doing this searching in a number of regions, and they're told in every region, there's a magical creature who's hidden their coin. Each creature has 3 hiding spots, and you have to figure out they'll hide more in their favorite places, and they'll hide fewer in their less favorite places. In each block, they have to learn through experience which hiding place is the most likely to be rewarded. Now, importantly, they do so over the course of 15 trials per block. Importantly, when they transition into the next block, they're told, because every magical creature has its own favorite hiding places, you have to learn anew where the coins are likely to be hidden. So all 3 of the stimuli they encounter in a new block are equally uncertain in their reward value at the start of the block. However, One of the three stimuli will be entirely novel. They've never seen it before over the whole course of the task. Two will be familiar, and they will have appeared in at least one of the prior blocks. And so in this manner, we can dissociate perceptual novelty and value uncertainty over the course of the task. Okay, so we can quantify three measures. Value, which we can model as the mean of a beta distribution that is distributed according to the reward history of wins and losses that the individual has encountered. Uncertainty, we can operationalize as the variance of that distribution. And novelty, which is inversely related to the number of prior exposures to the stimulus. Okay, so we can look at the effect of each of these measures on choices. So first, participants of all ages made value-sensitive choices. They're reward-seeking, and they are more likely to choose options that have a higher expected value, increasingly so with age. This is a common finding. Okay. If we look at how novelty influenced their choices, we can see that all of our participants are novelty-seeking. They're more likely to choose an option that has equivalent expected value if it's novel. If we look at the effect of uncertainty, we see increasing uncertainty aversion with increasing age. And I should note that this replicates the prior pattern of observed behavior in adults in a variant of this, in a non sort of child-friendly variant of this task upon which this task was based. And if we look continuously across age and plot these effects for equal expected value trials, we can see that while the preference for novelty is age invariant, the aversion to uncertainty increases linearly with age. We fit computational models of the learning process to participants' choices, where the learning process was formalized as though learning were conducted by a forgetful reinforcement learning agent, where every choice option is represented as a beta distribution that is a recently weighted integration of the win and loss histories. We compared three different models. One that allowed for optimistic initialization of the value of novel options. Then on top of this novelty bias, we added another free parameter that allowed, it was a free weight that allowed uncertainty to be weighted and summed with expected value. And a final model that proposed an interaction between stimulus familiarity and uncertainty, such that the uncertainty of more familiar options would influence their value to a greater extent. We found that while the choice behavior of children was best fit by the simple model that proposed a positive initial value for novel options, that adults and adolescents' choices was best fit by a more complex model that inflated or that decreased the aversive influence of uncertainty more so for novel stimuli. Okay, so here we found no evidence of developmental change in the influence of novelty on exploratory choices. Instead, our data suggests that heightened exploration early in development may be driven predominantly by a greater willingness to engage with uncertainty of choice options. One thing that we didn't find here that we're quite curious about, however, is, here we see age invariance in the effect of novelty and the linear effects of uncertainty on exploratory choice. We're seeing no hint of anything that is adolescent specific in this behavior. Most of the demonstrations that have found heightened exploratory behavior during adolescence have been looking at more complex behaviors in ecological environments. And so one question that we have is what properties of those environments that are not well captured by perceptual novelty or value uncertainty might be driving adolescents' exploratory behavior, and there may be several. Okay, so here I've told you, I've provided picture of exploratory behavior and the factors that might drive it and the consequences for socio-emotional behavior by bringing together measures of exploratory behavior in the wild with the study of exploration in the lab. Going forward, there are a number of directions in which we'd like to sort of push this research further. We're really interested in understanding the relationship between exploration and learning. Here we looked at social network formation, affective fluctuations, but you can imagine if you're looking at the behavior of an individual who is going about their day-to-day activities, some of the locations they're visiting are reflecting something that is akin to exploitation. There are known reward values to visiting certain locations in their environment. And at other times, they may visit novel locations, have positive experiences, and be more likely to return to them. We've actually recently collected data from 2 groups of individuals who have recently undergone life transitions, entering high school freshmen and entering NYU freshmen, many of whom are new to New York, and we got them on their first day in the dorms. So we're interested in looking at the evolution of their behavioral routines over time and seeing whether we can see the emergence of pattern as reflecting this sort of exploration-exploitation trade-off. We're also very much inspired by recent work from Hugo Spear's lab that has shown that individuals who grow up in cities with higher or lower degrees of entropy in their street networks differ in their navigation ability. And we're interested in looking at real world movement patterns in childhood and seeing whether this relates to the formation of spatial maps and navigation ability and examining this longitudinally over time. We're also interested in understanding the predictive significance of individual differences in patterns of exploratory behavior. We were really inspired by some work by Oren Forkash and colleagues looking at personality like dimensions of behavior this was in rodents in natural environments. So using these sort of deep phenotyping approaches that we've heard about in prior talks, there was an attempt to look for behaviors that were individually stable, but stable within an individual, but maximally variable between individuals. And we're interested in the idea that patterns of movement behavior may be reflective of personality-like traits, and are in the process of collecting data to further test that hypothesis. Finally, we're also interested in the relation between these exploration affect dynamics and the implications that they might have for understanding psychopathology. So there are disorders like depression that are associated with inactivity, behavioral rigidity, and dysregulated affective dynamics. And one of the most effective evidence-based therapies for depression is behavioral activation therapy, which really encourages people to get out and re-engage with their environment. And we're interested in testing the hypothesis that one element that may make those therapies effective is exposure to environmental novelty and experiential diversity. Okay. So I want to stop. I want to make sure I acknowledge the people who were centrally involved in this work. All of the geolocation work that I told you about was done in collaboration with Aaron Heller at the University of Miami. Natalie Saragosa Harris is a doctoral student at UCLA, former lab manager in my lab, and was the first author on the adolescent exploration study. Kate Nussenbaum, a current grad student in my lab, and Rebecca Martin, a former postdoc, led the laboratory study on novelty and uncertainty in exploration. I want to thank my current lab members, past lab members who were involved in the research, my collaborators, funding sources, and thank you for your attention.
We have time for a few questions, and we're going to try one more time with the other talk.
Hi, over here. Yeah, that was a really great talk. I had a question about the lab study where you saw a decreasing tolerance for uncertainty with age. And in the natural environment, right, with age, people probably experience less uncertainty as they have more and more sophisticated models of the world. So I wondered if you could tease that apart at all experimentally. whether tolerance of uncertainty is related to the extent of uncertainty that the individual is experiencing even in the test environment.
So you're asking a fabulous question. You know, we've created this artificial environment in which we can perfectly manipulate the level of uncertainty, but in natural environments, as we age, we understand much better the structure of our environments. And so we may be less likely to confront uncertainty, and moreover, it may be a reasonably adaptive heuristic to prioritize actions for which we have a confident high value, and to kind of shy away from prospects that that have uncertain value. We don't have any data to speak to this, so I'm really speculating. You know, I think the sorts of conditions ecologically that might reproduce a sort of that might engender a higher level of uncertainty in the natural environment might be things like moving to cross-cultural, whenever you leave the environment in which a lot of your learning has occurred and transition to a new environment. So I think those would be the right.
That'd be a good test case, because it seemed like it might be coming on a framing effect relative to sort of life experience uncertainty.
Yeah, absolutely. Cool. Yep.
Hello, that was super interesting.
Thank you.
So I just had there's a body of literature that's about the amount of exercise also correlates with positive emotion.
Yeah.
And I imagine people who travel farther and longer see more novel things. So did you look at the correlation between just pure mobility in your entropy measure, or how are these related?
I want to make sure I heard. So distance traveled, I think, is the confound that you're worried about. Okay, yeah. So all of these effects survive control for distance traveled. Your intuition, I think, is a good one, that when we travel over longer distances, we may see more things. The thing is that often when we travel over longer distances, we do so at higher speeds, right? In cars and trains, and in our data, we modeled. We were recording a GPS coordinate per minute. So, and we were agnostic as to the mode of movement through the world. So we treated walking and subway riding as though they all have equivalent potential for novelty. Or that they contribute to experiential diversity equivalently. So, even though you're traveling a further distance, if you're doing so at a higher speed, maybe it's not going to inflate entropy to that much greater of a degree. It might mean that you're seeing more sights and sounds, but we don't see that it's the case that distance traveled is producing effects on positive affect. Number of locations is strongly related both to roaming entropy and positive affect, but those were the kinds of effects that we sought to control for with things like our experiential diversity analysis, which that sociodemographic feature space really breaks down the relationship to number of locations.
So I know there's some more folks who'd like to talk to you, but hopefully we can do that offline. And I ask everyone to thank Kate again and welcome our final speaker.
Wow, what a whirlwind of a session. All right, I'll try not to eat too much into dinner. My name is Nathan. I'm a third-year graduate student in the Williamson Lab at the University of Pittsburgh. I'm going to talk to you about auditory-guided behavior, which essentially occurs anytime that a sound influences our decisions or actions. One example of an auditory-guided behavior is auditory categorization, which is the transformation of acoustic features into discrete perceptual categories. To give an example of this, if you're on the dance floor and a new song starts to play, you can quickly determine the tempo of that song, determine if it's slow or fast, and then decide how to dance along to the song. But how do we accomplish this? The auditory cortex is not a hub for motor control, so it necessitates that auditory information is communicated throughout the brain to influence things like our decisions, actions, and emotions. As a result, our lab is interested in understanding how the auditory cortex communicates with the rest of the brain. To do this, we study projection neurons within the auditory cortex, which can be broadly classified as either intratelencephalic, which project to the cortex and the striatum, or extratelencephalic, which project only to subcortical regions. And my research focuses on these ET neurons found in layer 5B. Recent work in our lab has focused on mapping the inputs and outputs of these ET neurons in the auditory cortex. We know that they send dense downstream projections to targets such as the thalamus, inferior colliculus, striatum, and amygdala, regions known to play roles in decision-making and action. We've also used monosynaptic rabies techniques to label the inputs selectively onto these ET neurons, and we find that they receive inputs both from auditory regions as well as higher cortical areas. So this anatomical organization leads to the portrayal of ET neurons as integrate and broadcast neurons, which pool inputs from multiple different sources and broadcast that information throughout the brain. So to study the role of these ET neurons in an auditory-guided behavior, we use a head-fixed auditory categorization task. So water-restricted mice are trained to discriminate between slow and fast rates of amplitude-modulated noise by licking either left or right. In a given trial, the sound plays for 1 second, and shortly after sound onset, the mouse is free to lick to make its choice. If it categorizes the sound correctly, it receives a small water reward, and if it categorizes incorrectly, it receives a 5-second timeout. Mice are initially trained on a simplified version of this task until they reach proficiency, and then they are asked to categorize all 9 stimuli. Mice are able to readily learn this task and continue to improve their categorization throughout learning, as reflected in these psychometric curves, with increasing slope and decreasing bias across days of learning. While the mice perform the full version of this task, I use 2-photon imaging to capture the activity of these ET neurons during behavior. GCAMP8S is selectively expressed in these ET neurons through a retrograde injection into the inferior colliculus. I can then capture the calcium activity of each neuron and then deconvolve it to get an activity trace more closely resembling spikes. So to start understanding what's going on in this ET population, we looked at some example neurons in expert mice. Here I'm showing 2 example neurons with trials on the y-axis sorted by which stimulus was played, and the color reflects their activity. What we noticed is there are certain neurons in the ET population that seem to exhibit tuning to either the slow or the fast category. And this suggests that individual ET neurons have categorical selectivity based on their training. So to quantify this, we used a categorical selectivity index, or CSI, where values near 1 or -1 would indicate strong selectivity to the slow or fast category, respectively. We can quantify this for the entire population and plot that as a histogram. And here I'm just showing 3 example time points in training. But what you can see is that early in learning, this distribution is centered around 0, suggesting not much category selectivity in the population. But as the animals improve at the task, this distribution spreads to a more bimodal shape, suggesting really strong categorical selectivity in this population. Furthermore, the amount of categorical information in the ET population on a given day actually strongly correlates with the success rate of the animal on a given day. Notably, when we have the mice just passively listening to the exact same stimuli, the same population of ET neurons no longer exhibits any categorical selectivity. So this suggests that ET neural activity changes across learning to encode these behaviorally relevant perceptual categories, while the tuning to stimuli outside of a behavioral context remains unchanged. Now, our task also contains an auditory stimulus at the category boundary, which is neither slow nor fast, but rather is rewarded probabilistically on either side. And this allows us to study the animal's choice irrespective of stimulus. So I used PCA to visualize the activity on these trials, and I'm coloring them based on whether the mouse licked left or right. And what you can see is that ET neurons show distinct activity profiles based on the choice the animal makes, regardless of if that choice was rewarded or not. So this suggests that ET neurons display choice selectivity even in response to identical auditory stimuli. To further quantify this choice selectivity, I trained a linear SVM to decode the animal's choice across all time points of the trial, and I noticed 2 changes across learning. First, the peak decoding accuracy increases from around 80% early in training to around 90% late in training, and the location of this peak actually shifts closer to the time of sound onset as reflected by the arrows on the x-axis. And this actually mirrors what we see during behavior, where mice that are better at the task have faster reaction times. So therefore, ET neurons increase their choice selectivity across learning. Now, I've shown you from a population level that these ET neurons have categorical and choice selectivity, but now to address what's happening at the single neuron level, I used longitudinal imaging of this population. And here I'm just showing 2 example neurons, but hopefully you can appreciate that these 2 neurons are actually dynamically shifting their tuning properties across learning to reflect these perceptual categories. I can show this for the entire population as well by plotting the CSI values for all matched neurons across days. And what you can see is that strong categorical selectivity strongly increases in the entire population, as reflected by darker colors towards the right of this plot. Furthermore, looking at just the passive responses of these track neurons, it does not seem to have any dramatic shifting in tuning across learning. Therefore, these ET neurons develop categorical selectivity through dynamically changing their tuning properties across learning in a task-dependent manner. But if you remember back to the beginning of the talk, I mentioned that there are these other excitatory neurons in the cortex as well. So how do we know that ET neurons are actually doing something unique? To begin to address this, I repeated my experiments in a separate cohort of mice, this time imaging in layer 2-3. And when I look at the CSI histograms for this population, I do not notice any changes across learning, and I only see maybe weak categorical selectivity. This is reflected in the matched neural population as well, with only a small fraction of neurons tuned to these categories, and these responses remaining stable across learning. We can look at the choice decoding accuracy of layer 2-3, and again, we do indeed see choice information in this population, but it was nowhere near as strong as in the ET population, and again, does not seem to change across learning. Finally, I wanted to look at IT neurons specifically in layer 5 to see if this was a general layer 5 phenomenon or if ET and IT neurons are actually doing something different. And when I look at the CSI histograms of this population, I noticed something surprising, where across learning, this distribution actually seems to become more centered around 0, which was the opposite effect I saw for the ET neurons. And this would suggest an actual decrease in categorical information across time. This is also reflected in the matched population, with the strongest CSI values appearing early in training. So this loss of information is even more apparent when we look at choice decoding accuracy across learning, where initially this population is decoding at a peak of around 85%, but by the time these animals have been trained on the task for multiple days, that decoding accuracy has significantly dropped to around 65%. And this drop in selectivity is still something we're actively investigating. But one potential hypothesis is, given that ET neurons increase their selectivity across learning and layer 5 IT neurons decrease their selectivity, it suggests that there could be some sort of trade-off of information between these 2 populations. So in conclusion, ET neurons increase their selectivity for both stimulus category and behavioral choice across days of learning. And all of this together suggests that ET neurons play an important role in transforming acoustic features into discrete perceptual categories. With that, I would like to thank all of the members of my lab, especially Megan, who did the anatomical experiments, Becca, who kind of pioneered this behavior in our lab, and of course, Ross for all of his guidance. Thank you so much.
Thank you, Nathan. We have some time for some questions.
Hi. I was wondering if you have an idea what drives the shift in the IT and in the ET cells.
Yeah, that's a great question. I don't know if there is some kind of top-down mechanism that's changing like gating of what's relevant and what's not. If the ET neuron itself is becoming more recurrently active, I can only really speculate.
And IT cells are also strongly connected to the ET cells. Do you think it's like a shift of the information then from IT to ET?
Yeah, so that's a good point. The reason I say that is because IT neurons project to ET neurons, but there's no recurrent connections from ET to IT. So if there is some kind of passing of information, it is one directional.
Thanks.
Thank you.
Okay, well, thank you very much, Nathan.
So thank you, everyone. We're very sorry for the tech issues we had in this last session today. Sometimes fate just isn't kind. Speaking of which, don't forget that tonight is daylight savings. Have a good evening, though. Go have some fun. Enjoy the poster session. Enjoy the